Hey guys.
Hey everybody. We will get started in a few minutes.
How's everybody doing? I've seen a lot of you guys stressing a little bit. Don't worry.
Yeah.
We were the same way. We were the exact same way before this exam. I will tell you that the week leading up to this exam, I had a few legit panic attacks because I was like, I'm gonna fail this exam. It's gonna be terrible. I did a lot better on DM than I thought I did. And we took it close to a weekend. And so, it's 4 business days before you get the exam back. But since it was a weekend, it was 6. And I kept dreaming that I was getting my exam back. And every single time I dreamt it, my score got lower and lower and lower until I had convinced myself I had failed the exam. And I did a lot better than I thought I did. So if anybody wants to take any sort of solace from that story, you can. But we understand how stressed you guys are.
Yeah, for sure. It's a lot. We're gonna point out the stuff. It's one of those things, if you like whiteboarding, good for you, but no judgments. But we're gonna point out the enzymes you need to know that will be tested. Unfortunately, because of our schedule, we had to compile this into one review. Last term, we did it in 3 reviews and DM over 3 reviews, and it was over 7 hours. So, so what we're gonna do now, if you want to go back and look at those reviews for more of a comprehensive look at the material, then you can go ahead and do that. But what we're gonna do now, just so that we're not here nobody's here for 7 hours, we're just gonna kind of point out the stuff that's really high yield. Not a lot of necessarily teaching or explaining, but it's more of this is high yield, this is high yield. Because at this point, I mean, y'all are pretty much good to go. You just need to maybe fine tune a few things in regards to the material. All right, so I will share my screen. And we compiled all of the slides, and it was 600 slides. So yeah, again, we're just going to point out the important stuff for you guys. Right. Okay. And by the way, because things have been said, the reason we rebranded, the only reason we rebranded, so you guys know, is that the school requested we do that, just because we're not running official PLGs anymore. So we're not this is not a big scheme to start making money or whatever. The school asked us to take the DES off of our name, so we did it. Okay, so that's the only reason we went through that process. But you could find our pages there. As always, our additional resources are posted. So Lindsey's going to cover the metabolism. I'm just going to do a little brief overview real quick. So some of this stuff's from first aid. I love first aid. Kind of just simplifies everything and puts it, not too much explanation, but good high yield facts here, right? So kinases always add a phosphate group. Phosphatases always remove a phosphate group. And so of these, those are the most important things to know. Another good trick, carboxylases always use biotin as the cofactor. So sometimes they ask that if they ever ask a carboxylase reaction, biotin's always going to be the cofactor. All right, so this is kind of the breakdown, what we're going to start talking about here. And again, of this, really the only true pathways you need to be able to write out are the glycolysis pathway and the TCA pathway. I didn't go into this exam being able to write out all this stuff. But if you ask me, of the ketosis pathway, right? What is the rate limiting enzyme? I could tell you. Or if there was a specific disease-oriented step in a pathway or a drug target, I could tell you that. But by no means could I write this whole thing out. So don't stress on that. You want to focus on the things that can be tested. They're not going to ask you some enzyme that's insignificant, right? They're going to ask you things that make a difference. So one of the key things when you talk about rate limiting enzymes, they're always unidirectional, right? So the ones that are bidirectional, those are the ones that tend to be that don't tend to be important. The reason the rate limiting enzymes are unidirectional, or put it this way, those unidirectional enzymes by definition make them rate limiting for these steps, because it's like a turnstile, right? Like you're either going to have high blood glucose and you're going to want to break it down or get it out of the blood, or you're going to have low blood glucose and you're going to want to mobilize everything and get it to the blood. Okay, so it's all or nothing pathway. So these unidirectional enzymes allow you to go one way. If everything was bidirectional, it would kind of be fuzzy, right? Like sometimes you're going to want to go this way, and so you need to be definitive, right? The body's definitive. It's like, okay, now we're going to release insulin. We have high blood glucose. This is what we want to do. So everybody move this way. So those turnstiles or those unidirectional enzymes allow you to move in a single direction. Okay, so we'll get to some of those in a second. So this is a super important slide because they're going to give you questions on the exam, and it might not be so straightforward, but they'll ask you, they'll say, they'll give you some scenario where you want to definitively say, so like it'll be a second order question, and you'll have to determine that you want to go through fatty acid synthesis, right? In this pathway, you'll say, person just ate, they want to store their energy, right? So then you would determine that it's fatty acid synthesis, and one of the selections or the answer choices will be acetyl-CoA carboxylates, right? So that's the idea. You want to be able to say, okay, from the question, you want to say, this is the pathway we're going to use, and they're going to ask you what is the most important enzyme in that pathway. Okay, so I'm sure y'all came across some of those practice questions like that, but that's what they're going to ask. They're not going to ask you, obviously you won't have to write out the pathways or anything like that. They're going to ask you, what is that rate limiting enzyme? What is the most important enzyme in the pathway? Of course, they also could ask you these regulators. Some of them are straightforward. Obviously, if you have a lot of ATP, then you're not going to go through the process of making more ATP, right? You just don't need to. Whereas if you have ADP and you want to make more energy or more ATP, then you're going to go through the process of the TCA cycle. Okay, so use that to your advantage. But some of these, typically those proteins are the ones that mess me up, like citrate and stuff like that. So those are the ones I try to highlight or go through because it's not exactly straightforward. But yeah, so this is all fair game as well. I use this idea, at least I tried to last term, to do this on-off switch. So this is the idea of saying we're going to go one direction or we're going to go the other way, right? So in the aspect of just eaten postprandial, insulin is going to be secreted. You want to take that glucose out of the blood and mobilize it. You want to get it to the liver so you could store it. You want to get it to the muscle or the brain so you could use it, right? So this is the idea of the on switch, right? The fed state is the on switch. Okay, so remember that you could think of the idea of dephosphorylating all these enzymes as saying we're flipping the switch on. That is the marker for all these enzymes to go through. If the enzymes necessary for mobilizing the glucose out of the blood are dephosphorylated, then that's the process of turning them on. Okay, so that's kind of how you tell the enzymes through this pathway that insulin is being released. We want to mobilize all of this blood glucose out of the blood and store it. Okay, so the opposite's true for glucagon. It's off. So these enzymes that are necessary for bringing glucose back into the circulation, mobilizing glucose into the blood, that'll be the phosphorylation step. Okay, so remember, kinases will phosphorylate, phosphatases will dephosphorylate. There are very few things in medicine that are absolute, right? There's always gray areas, but this is one of those things. You definitively need to say we're dephosphorylating these enzymes so that we can definitively say we're going to mobilize everything out of the blood. We're getting the glucose out of the blood. Okay, so it's a very definitive thing. Okay, so they could ask you this. Don't get tripped up on it. There's no crossover. Either you're going to mobilize it with insulin or you're going to make more glucose with glucagon. Okay, so keep that in mind going forward because all of these pathways utilize that scenario. All right, so another important step in the process of making ATP, you have to use ATP. So remember hexokinase being in the muscle, glucokinase being in the liver. So these are going to be require ATP. And then of course, substrate level phosphorylation, that's the process of making ATP. So these are going to be the enzymes that do that. Okay. Again, like I said, these unidirectional rate limiting enzymes are going to drive the pathway in a single direction. Okay. Now, when we talk about gluconeogenesis, gluco making or the formation of new glucose, right? That's what we're saying there. So this is going to be, as you can see, we're off, right? So these enzymes are going to be phosphorylated, right? Because that's going to tell them to be turned on because we want to go through the process of making new glucose to increase the blood glucose, right? Okay, again, same process. These are all unidirectional enzymes. It is important to note, again, we said carboxylases use biotin, but this pyruvate carboxylase is in the mitochondria. Okay, so you need to know that because it uses this shuttling pathway to get everything out. All right, now everybody gets tripped up on this. We're going to break it down. It's this, and you know, you may ask yourself, like, what is the purpose? Like, why even go through this process of having this bifunctional enzyme? And the way I think of it, it's kind of like fine tuning this process. Like, you know, you have the single pathway, but if you do this, you can even make it even titrate even further. So what I did is I broke it down like this. Now, again, the light switch is on, we're dephosphorylated, okay? But it's tricky because we're actually dephosphorylating these kinases, okay? So these kinases are going to add a phosphate group, but they're dephosphorylated, which means these kinases are active during the process of lowering blood glucose, okay? So don't get tripped up like that. The kinases are gonna add phosphate groups, but they had to be turned on by the dephosphorylation process, okay? So it's a little trippy, but think about it like this. So our unidirectional pathway, we have blood glucose, high blood glucose, insulin's released. We want to go through the process of getting rid of the glucose in the blood. You want to go through glycolysis, right? So anytime you have high levels of fructose 2,6-bisphosphate, this is that bifunctional enzyme, or this is that process of the bifunctional enzyme upregulating fructose 2,6-bisphosphate, okay? So the easiest way to think about this is who's on what team, okay? Fructose 2,6-bisphosphate, phosphofructokinase 1, phosphofructokinase 2 are all on this pathway or all on insulin's team, right? So we're trying to lower blood glucose. So anytime these are elevated, we're going to go through the process of glycolysis, okay? Now, if you look at the other team, fructose bisphosphatase 1 and 2, they're on the team of glucagon, right? So we want to basically make new glucose and mobilize it to put it in the blood, right? So they're on the other team, which means that if that's the case, then low levels of fructose 2,6-bisphosphate are going to push this way. So that's the trickiest part about it. You need to ask yourself, if we have elevated levels of 2,6-bisphosphate, what are we doing? That means we're going through the process of glycolysis. If we're doing gluconeogenesis, this is going to be downregulated, fructose 2,6-bisphosphate. So if it's still confusing to you, just remember who's on what team, okay? Because that'll tell you which direction you're going to go. All right, the same thing here. We use the light switches. So anytime dephosphorylation or phosphofructokinase 2 are upregulated, then we're going to go through the process of lowering blood glucose. And the opposite is true here. Phosphorylation, fructose bisphosphatase 2, and this is going to go through the process of upregulating or bringing blood glucose up, your blood glucose up, okay? Right, very, very important, super important. They use the mnemonic TLC for Nancy. These coenzymes, they ask this on every exam. These 5 coenzymes kind of go together. And I put down here what enzymes or what pathways that these 5 enzymes are used for. Pyruvate dehydrogenase, alpha ketoglutarate, which are the ones that are maybe straightforward for you because they go through the Krebs cycle, but also this branch chain keto acid dehydrogenase is also another one that uses it. So you need all 5 of these cofactors to go through this process. Okay, so they like to test on that. And you can see here, if you're going to whiteboard something, this would be what to whiteboard. Knowing the coenzymes that are necessary, knowing the enzymes that are necessary, and knowing the process, even knowing GTP comes through this pathway here, where you're going to make NADH, FADH2. Okay, so for the rest of the pathways, I think it's a lot lower yield, but for this one, I think they could ask you any of these. Sometimes they give those questions where they don't put any of the enzymes or the substrates here, and they just put numbers or letters, and they ask you what enzymes are used there. So fair game for this, make sure you write that out. And then this is the process. So you should know this too, right? And for this, right, the electron transport chain, these drugs are super important. Easy points for you, just know which ones go with which complex. Other than that, you should be fine. Sometimes, I'm sure I have it somewhere. Yeah, these, okay. So yeah, definitely know which drugs go with which, but there's also some that come in later that put pores in. Yeah, these, know how these work as well. I don't know. And these right here, yeah, know these and how they work. They basically punch holes in the membrane so that the hydrogen ions can pass through. So make sure you know these. They're similar to oligomycin, which is the complex 5 right here, the ATP synthase, right? So remember complex 5, that ATP synthase is turnstile, or I know I use turnstile again, but it is actually a turnstile. Like as the hydrogen ions come through, it turns and it allows the process of making ATP. Okay, so just kind of know how these drugs work. It should be easy points as long as you know their mechanism of action. These uncouplers, DNP and aspirin, work like that. DNP has been used as like a weight loss drug, but the idea is that if they punch holes in the membrane, then the hydrogen ions just pass through. Okay, so the energy is burned as heat instead of actually making ATP. So it's the same concept as brown fat that you see in infants and like bears when they hibernate. So this is an uncoupling process, but all you really need to know is that it puts a hole in the membrane. So those hydrogen ions it's not an efficient process of them going through that complex 5 turnstile, right? It's just burning through heat. Okay, so it's a way to burn energy opposed to making it. So make sure you know those. These as well, these ionophores, these are the ones that form these channels. Yeah. All right, and then, right, so glycogenesis and glycogenolysis, right? Okay, the process of making glycogen is the idea of storing glucose. It's just a bunch of different molecules. So knowing which enzyme goes with which is important, but even more important is knowing the diseases that go with each of these processes. So we're going to talk about this a little later. These diseases that go with which, because as you can see here, some of the enzymes can actually, I think of it like it's like Pac-Man, right? So they can eat away at this, but once they get to the glucose where you get this branch point right here, you need to use a different enzyme. So this is the concept of the limit dextrins, but we'll talk about it when we get to these glycogen storage diseases. Okay. All right, I think that's that was just the overview. So Lindsey, you can take over and share your screen. Whoops. So Lindsey's going to cover it in a little bit more detail. I just kind of wanted to give you a little overview on that.
I'm trying to get the PowerPoint to cooperate with me. Okay, here we go. Oh, wow, no. Brady, when I'm trying to present this, it blows it up really. So I'm just gonna scroll through it. I'm not gonna waste you guys' time trying to figure it out. Here. Okay. So I'm going to go through this as quickly as possible with giving you guys the big points you need to focus on. Hold on, I lost my place when I was playing with it. Here we go. Okay, so I do recommend drawing it out, your 24 hours before the exam. So there's not a whole lot you can do at this point if you haven't done it already, in the sense of drawing it out. But like Brady said, you don't need to memorize every single detail of every single pathway. There are things that you need to focus on for each, and we're going to go over this. Ninja Nerds was my biggest thing. So glycolysis and the pentose phosphate pathway, general facts, know the main idea, energy production, ATP, it's in the cytosol. So this is your summary. Know which levels are substrate level phosphorylation, which are regulated, so they go hand in hand. So the reason why this is important, Brady already touched on this, is because if something goes wrong in this step, it can lead to disease, and it can lead to an increase of intermediates or a decrease of something afterwards. And so, for example, if a question that they've loved to test us on since we started science is, you block enzyme B, what happens? A will increase, like substance A will increase, substance C will decrease, that kind of thing. So just understand that general concept. Glucokinase, hexokinase, this is very high yield. It'll be 1 or 2, maybe 3 questions on this. So glucokinase is insulin dependent, sorry guys. And then the mutations will lead to hyperglycemia and the MODY2. Yes, no, the KM and V max for glucokinase versus hexokinase, that's something that they do like to focus on. Okay, so fate of glucose, 6-phosphate. So this is after glucose gets trapped into the cell. This is glucose trapping, converting it to G6P. You have a few different places it can go. Glycolysis is just one of them. You can also do the pentose phosphate, and you can also do glycogenesis. All of this depends on what the energy levels are like in your cells. So if you have a certain amount of glucose in your body and you need storage, you'll go to glycogen. So it all depends on hormones and different regulators. And so knowing the different ones for each is important. Pentose phosphate pathway. This slide pretty much has everything you need on the pentose phosphate pathway. It's not anything too complicated. The big enzyme G6P dehydrogenase forms NADH, purine pyrimidine synthesis, and then transketolase is the one that requires vitamin B1. Yes, please know the different ways they can call it. So TPP versus vitamin B1, they can ask either of those, but those big points, it's basically everything you need to know about the pentose phosphate pathway. The bifunctional enzyme, this is kind of confusing, but once you understand it, it kind of simplifies it. This is a little sticky note I made myself to simplify it. So glucagon phosphorylates both PFK2 and F26B, biphosphate. The difference is that this phosphorylation is going to do something different to each enzyme. So if you have phosphorylation, then PFK2 is going to be inhibited, and then F26 bisphosphatase is going to be activated, and then insulin is going to dephosphorylate. So when they're dephosphorylated, it does something else. So this is the big idea here on this little orange sticky note that I have. So I wrote it out a little bit over here, but the regulation is going to be the biggest thing on that. Okay, this is kind of more of the same. So the bifunctional enzyme can be phosphorylated by pKa in the liver. Which of the following are, oh, it's a question. Okay, you can, I'll give you a few seconds to answer this. Remember, privately to either Brady or myself. Yeah. So there are two, phosphoglycerate kinase and pyruvate kinase. Notice how we emphasize the word kinase, because substrate level phosphorylation are going to be those kinases. So this is just the formation of ATP. This slide is just focusing in on that. Yeah, I'm not going to read that to you. Fate of pyruvate, do be familiar with all of these, because each one of these is going to have a different condition to go there. So lactate, the lactic acid cycle, that's dependent on NAD versus NADH, and alcohol can really affect that. And then oxidative decarboxylation, going to acetyl-CoA to go to TCA. Oxaloacetate, this is the one I believe that's involved in the gluconeogenesis. Right here, don't quote me on that. We're going to go over it later. I'm trying to remember it just on the slide. I apologize. And then transamination, this is the big one that comes up later. Okay, so regulation, again, regulation is big, 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 big. So feed forward activator is the fructose 1,6 bisphosphate. Why? This makes sense, because feed forward is something that is telling something along the line, hey, get ready, hey, they're coming. So if you have a buildup of fructose 1,6 bisphosphate, because you're going through glycolysis, everything downstream needs to start getting ready, because they're going to get the byproducts of whatever came before it. And so this is why it's a feed forward activator. It's saying, hey, the system's working, you need to be prepared, because you're going to have to do some work in a minute. So that's what it means. And then phosphorylation decreases, alanine inhibits. So know those regulators. Okay, this is a very long slide. There are like two things that I want you guys to take from this: NADH, NAD ratio. So lactate. So that this is very important. And then just know the fate of lactate. It goes into the Cori cycle, because when you're exercising, you are going to build up lactate, of course, and lactate through the Cori cycle is going to be able to be recycled for energy. That's why, because in the muscle you're releasing lactate, and then it can be used for gluconeogenesis in the liver. So you work out, you release lactate, goes into the bloodstream, lactate goes up to the liver, it can be used for gluconeogenesis. I don't know why this was such a hard concept for all of you. I don't know why this was such a hard concept for me to grasp, because it's such a simple concept, but it took me so long to understand how simple the Cori cycle was. So that's basically it. So anaerobic glycolysis, of course, formation of lactate, especially in the skeletal muscle, goes to the liver, gluconeogenesis. Okay, comparing energetics. Energetics of glycolysis. Oh, I remember why I had this in here. So just knowing the difference between aerobic versus anaerobic, there is a slight difference. No difference, maybe one question on your exam, but just be aware of that. Defects in muscle glycolysis. This is okay. This is big when we talk about disorders of glycogen metabolism, all of that stuff. So during high intensity, this is extremely, extremely, extremely high yield muscle cramps during high intensity aerobic workout. We're not talking about the diseases right now, so I'm not going to go too in depth about that. We'll do that in a minute. So this is an introduction into later talking about the glycogen storage diseases, but just understand that the presentations of all of those diseases are extremely high yield, because you want to be able to differentiate in the clinical vignette so that you can get the answer. So that's why I say that. Arsenic poisoning, just know the enzyme. Lactic acidosis creates a high ion gap metabolic, so high anion gap metabolic acidosis. Just know that they are associated with one another. And then these are conditions that can cause it. They love to associate the alcohol relationship, NADH to NAD ratio, to this. But just understand that there are many things that can cause this. Regulation of glycolysis. Did someone say regulated enzymes? So glucokinase, PFK1, pyruvate kinase, know how they are regulated. Structure and function of carbohydrates. I'm going to go through this pretty quickly. There are a couple questions. Reducing sugars, just understand that there are reducing sugars, and then reducing sugars in the urine. You'll see, you probably have seen in your IMCQs and ESOFT quizzes, like they're reducing sugars in the urine, stuff like that. So just understand that that is a thing. So I'm gonna piles. This is really big. Sorbitol versus galactosol. Understand the difference between these two. They are both highly osmotic, and so sorbitol, galactosol, they draw in water. That's why they cause cataracts. That's why they cause destructive things. But no, because galactosol is from galactose and sorbitol is from glucose. So know that it's different. So diabetes mellitus versus galactosemia. Galactosemia is something that they talk about in another DLA. I think it was a TROTS one. But just there are a couple kinds of galactosemia. Yes, please know the difference between the couple kinds of galactosemia because they will test you on it. But these both are high yield points. I would know those very well. The next couple ones, lactose, sucrose, and then maltose. These are both all brush border disaccharidases. Know that term, brush border disaccharidase, because they could, instead of saying lactose or sucrose or maltose, call it a brush border disaccharide. So just understand that. And this is a big thing. If you have something wrong with the brush border, you don't get this absorption. So lactose intolerance and stuff like that. So that's the big thing here for these three sugars. And then if you're getting down to crunch time, and so maybe if you have time, just understand the alpha 1,4 versus like beta 1,4. They might use that. Glycogen, liver and muscle as glycogen granules. This is where it's stored. So you learn more about this when you actually talk about glycogen synthesis, the alpha 1,4 or alpha 1,6 linear versus branch. So that's going to be big later. Starch, I do remember something about either starch or cellulose. So I would understand the alpha 1,4 versus alpha 1,6. And then cellulose, dietary fiber, unbranched homopolysaccharide. If they're giving, I do remember on this exam, if they give you an alternate name for something, so for example, cellulose, unbranched homopolysaccharide, that it was kind of big on this exam to use the alternate term. So just know both of them. Bioenergetics, there are a couple points here. This was a very long DL, I think it was a DLA, but there are only a couple points that you really need to focus on. Please do not memorize every slide of this. Now, it is big that you understand the difference between exo and endothermic. Know all about them. There are a few different definitions of graphs in that DLA on it. Please know all of them. Hopefully you have learned this in chemistry classes in the past, and so it will just be a refresher. But don't take this for granted if you haven't reviewed it yet. That's something that I do sometimes, is take a piece of information for granted because I've learned it in the past, but you need to bring it to the forefront of your memory. So those of you that are like me, please bring it to the forefront of your memory. It's an easy point if you can remember it. And then making an unfavorable reaction favorable. This is the idea behind adding ATP, because if you break that ATP, that last phosphate to phosphate bond, you get enough energy to overcome an unfavorable reaction. That's why some reactions require ATP to go, because it's on its own an unfavorable reaction. So that is basically the concept that we're going for here. Yes, you need to know how to do the calculations. Yeah, so it's called thermodynamic coupling. You need to know how to do the calculations forward and backward. If you spend a little time on this DLA, it's not very hard at all. And so if you are given, plus for, I think I have, oh, I don't know if I put one of the equations on here, but do please go through that DLA, spend up 5 to 10 minutes knowing how to do the reactions both forward and being able to recognize that is this a forward reaction, is this a reverse reaction. You will get a question or two on the exam. Okay, metabolism in fed and fasting. I'm not going to go through this. I put this on here for completion's sake. If you've gone through all of the other lectures, this won't come as a surprise to you. It basically just summarizes it for you. So I'm not going to read this to you because it's just 2 slides of bullet points, but do understand the difference between fed and fasting, what your body goes through, and just try to relate it conceptually, because a lot of this makes it fits into the bigger picture. And once you fit it into the bigger picture, you won't necessarily have to memorize that this goes with this. It'll just make sense. Okay, PDH and TCA. The PDH, this is that big enzyme that is the kind of bridge between glycolysis and TCA. There are 3 parts to it. I don't remember having to memorize the different parts of the enzyme, but what is huge are the cofactors associated with each individual one. This is my whiteboarding. You all probably know I love whiteboarding by now. So I whiteboarded everything out. So TPP is with the first enzyme. I forgot which one is here, FAD, but you need to know which goes with which. They will ask it. And then lipoic acid, thank you very much. And then you need to know what regulates it, of course. So regulation, so high energy charge off, low energy charge on. Know this circle, I think. So if you, PDH kinase is going to phosphorylate the PDH, and it's going to cause it to be inactive. So there are a few things that are going to influence this. So ATP, acetyl-CoA, NADH is going to essentially turn us off. Why? Because if you have an excess of ATP, if you have an excess of acetyl-CoA, if you have an excess of NADH, it means that the TCA has been working for a while, right? So if the TCA has been working for a while and you're getting all of these products, well, it needs a rest. You don't need to keep turning it on. So that means you can turn it off. So how are you turning it off? Well, you are activating PDH kinase, which is going to phosphorylate the PDH and make it inactive. So it's basically saying, stop. We have enough. Stop. Conversely, pyruvate, so an increase of pyruvate, so the end product of glycolysis, it's building up, and it's building up, and that can turn it on. That can inhibit this process. It'll inhibit the phosphorylation. So understand that. So kinase inactive, phosphatase active. So need to know PDH phosphorylate off, PDH dephosphorylated on. I hope I didn't talk myself in a circle there. I apologize if I did. Okay, we just talked about this, so I'm not going to go over that. Tender loving care for Nancy. This is the one that everyone likes to attribute to this. Please, 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 please know this. There are a couple enzymes that are going to use tender loving care for Nancy. Know them. So we have PDH. Does someone know real quick in the chat what's another one that uses TLC for Nancy? It's in the TCA. Alpha ketoglutarate. Yep. So know them again. I'm going to harp on this because this is something that tripped me up on a couple questions. Thiamine pyrophosphate is from B1. FAD is B2. NAD is B3. Please know what vitamin it's from. So vitamin B3, NAD. Please, please, please know that. Okay, so if you have low PDH, here are a few things. So Wernicke-Korsakoff syndrome, this is wacky, wild, and wobbly, I think they call it. And so that's just the clinical presentation: ataxia, ophthalmoplegia, memory loss, cerebral hemorrhage, confabulation. So wacky, wild, wobbly, you can write that next to it if you haven't heard that already. So alcoholics and malnourished. So a patient is going to be coming in and they're going to have a long-standing history of alcohol abuse, and they're going to be acting really crazy. They're going to be walking really weird. And so what it's going to be in reference to is a thiamine deficiency, PDH inactivity. Then beriberi, wet versus dry. So based on presence of edema. And then other thiamine requiring enzymes. We talked about alpha ketoglutarate dehydrogenase. Sorry, guys. But this is just because it's one of the coenzymes. And so if you have a decrease in the coenzyme, you're going to have a decrease there as well. But the one they always love to focus on is Wernicke-Korsakoff or beriberi. Okay. Again, if they give you a clinical presentation, please don't blow over this. Know that this is how they're going to tell you that this patient has a PDH deficiency. So a kid is going to come in and on physical exam, they're going to have frontal prominence, wide nasal bridge, flared nares. And so this is what's going to tell you that they have a PDH deficiency. I put this on here for completion's sake. I think I remember maybe one question on it, but just understand that there exists heavy metal poisoning and it can disrupt PDH. Okay, so regulation, I put all of this on the same slide. So regulation of citrate synthase, isocitrate dehydrogenase, and alpha-ketoglutarate dehydrogenase. This is one of the pathways that I do highly recommend you know every single step. The reason being it helps orient you to what part in the pathway you are at. So they will ask you about different enzymes here. They will ask you about different products. And so, you know, if this enzyme is inhibited, what can you expect? And so that's the thing where, you know, you'll have buildup of something, but you'll have a decrease concentration of something else. So this and glycolysis and the urea cycle maybe are the ones that I would suggest you knowing inside and out, the order of everything. The other ones though, just stick to the regulated enzymes, quite honestly. And then regulation. Oh my goodness. Hold on. My computer isn't. Okay. Got unplugged. Sorry. Okay. Regulation of citrate synthase. All regulated ones are irreversible, of course. Inhibited by its own product. Why is it inhibited by its own product? Because if you have enough of it, you don't need it to turn on anymore. So just understand that. Activated by substrate availability. So the big thing here is, do you need it or do you not need it? It's just yes or no. Whereas other things can come in that aren't not directly associated. So isocitrate dehydrogenase, allosterically activated by ADP and calcium. ADP, because of course, if you have high ADP, you are low in ATP. And why calcium? Well, where does calcium come from? Why would you see a lot of calcium being released? You can put it in the chat. Muscle contractions. So you're using muscle contractions, which means you need ATP. And so in excess of calcium can tell the TCA, hey, turn on, we need more ATP. So that's why it's an allosteric activator of isocitrate dehydrogenase. And then of course this isn't product inhibition because this isn't a product of this enzyme directly, but this process creates ATP and NADH. And so if you have too much, it's like, no, stop, turn off. We don't need you anymore. So that's the big thing here. And then alpha ketoglutarate dehydrogenase is a big one, like with PDH, uses TLC for Nancy, activated by calcium. We already talked about why calcium would be an activator in the TCA. Inhibited by NADH, we already talked about that. And succinyl-CoA, which is in the TCA. So that's a product of the TCA. You can read this on your own. I don't remember this being huge, but just understand that this exists. ETC and oxidative phosphorylation. So electron transport chain. I'm not going to read all of this to you, but you guys know that it creates NADH and FADH2, which goes into the electron transport chain, or TCA creates this. And then it goes into the electron transport chain, and it's the movement of electrons down all of these that creates the hydrogen ion gradient that causes the ATP synthase to make ATP. Yes, you need to know the different ones, and you need to know how the electrons are moving and what is associated. So FMN, you need to know this is associated with complex one, my God, complex one. So NADH dehydrogenase, so it's giving it to CoQ. And then complex 2, succinate dehydrogenase. Where have you also seen succinate dehydrogenase? This is kind of a small high yield point, but they do like to ask about it because succinate dehydrogenase is an enzyme in the TCA. It's just bound. It's a bound enzyme to the membrane for the TCA. And so that is a high yield point. So know where NADH gives its electrons to, know where FADH2 gives its electrons to, they give it to CoQ, and then how are the electrons getting to the third one, and then just understand this process, please. Yeah, so the big thing here, you're just creating the proton gradient because the proton gradient is what the ATP synthase is going to use. That's just saying essentially the same thing. ATP synthase, yes, know the difference between F naught and F1. You need to know that F naught is going to be it's in the inner mitochondrial matrix. And then the catalytic activity, highlight this. Catalytic activity is an F1. That's big. They might ask you about that. They like that. Okay, so that's the big thing here. You know, protons that have been pumped via the ETC are going to re-enter, and this re-entrance gives.
I know you know this, but can you tell me what you said we need to know?
Yeah, that F1 in the mitochondrial matrix is the catalytic activity of ATP synthase.
Okay, thanks.
Yeah, that's a big point, knowing which part of it is going to be the one that actually does it. And then it uses the proton gradient set up by the ETC to kind of churn itself, and then you're getting the ATP. So that's all structure. This ratio, I would understand it. Okay, I put a little star by this. So NADH, you're getting about 3 ATP at the very end versus FADH2, you're getting about 2 at the very end. So just understand that little point because they might ask about it. Now, NADH shuttles, you have to have a way for this to get across the mitochondrial membrane because it cannot pass. So you need a shuttle. So know the 2 shuttles, know what is involved in the shuttles because you have to get it across there. And this is how you do it. I would say medium yield for this, but don't underestimate it. Please know the difference between the malate dehydrogenase shuttle and the glycerophosphate shuttle. So ETC active when you have an excess of ADP, that makes sense because that means your ATP is low. Same over here. If you have a lot of NADH, it's going to turn it one way or the other. Inhibitors, very, very, very high yield slide, but it's also very easy points. Note every single one on this. Highlight all of them down here. So amytal is a barbiturate, rotenone is an insecticide. If you like Anki, they did a really good job at teaching me this. So rotenone 1, complex 1, pyricidin, it's an antibiotic. So that's that. Anti-mycin. Yeah.
We have a request. Can you go back to the shuttles real quick? I just want to, you really don't need to know much about it. I think the main thing is, so people wanted to just look at this real quick. The main thing you want to know is you actually get one more ATP through the malate shuttle than the glycerol phosphate shuttle. But other than that, like, you really don't, if they ask anything, they'd ask which one produces more ATP. So don't get too bogged down on that.
Yeah, I agree. Back to this, and then so anti-mycin A is complex 3. Complex 4, it's all the ides, so cyanide, azide, hydrogen sulfide, carbon monoxide. So all of these are going to be 4. And then oligomycin is the actual ATP synthase complex. Please know all of them. Like I said, high yield, but easy points if you remember them. Okay, so what's the difference between inhibiting the ETC and the actual ATP synthase? Well, if you inhibit ETC, you're inhibiting the flow of electrons and the proton gradient. So the proton gradient is messed up. You don't get that proton gradient. And without the proton gradient, you don't get ATP. So you are going to have an immediate reduction of ATP. So reduction ATP synthesis. Now, conversely, if you're inhibiting ATP synthase, but the ETC remains intact, the hydrogen gradient remains intact. So initially, you're not going to have something affected because electron transport, it's still going to happen. Now, too much of a good thing is a bad thing, right? You're going to get a maximum gradient because at some point you can't put more hydrogen ions in a room. Like you can't stuff more people in a room than can fit in a room. So at some point, this hydrogen ion gradient is going to max out. So when this maxes out, it can leak back. When it leaks back and it's not associated with ATP synthase, that's uncoupling because the hydrogen ion gradient, it's only supposed to be for the ATP synthase. That's it. Once the hydrogen ions are put into that matrix, they're supposed to stay there for use of the ATP synthase. But if you aren't making ATP, that machine is broken. ETC keeps churning out hydrogen ions into this gradient. It's like, okay, we can't fit any more hydrogen ions in here. It leaks back out irrespective of the ATP synthase, and so it uncouples. This gradient is now uncoupled from the ATP synthase, and you associate it with thermogen, and so this is not a good thing. But yeah. Okay, this transport, just know it exists. So adenine nucleotide translocase. So it's a symport. So it's going to, it's coupled to the other transporter. So a phosphate and a hydrogen ion. There are two things that can inhibit this. I do remember a question on this and I blew over this and I had to sit there and it took a lot longer to get this question than I wish it did because I didn't focus enough on this slide. They'll ask you about it, but it's not like the most high yield thing, but please don't underestimate the slide. They will very likely ask you a question on it. So please know the difference between these two. Okay, why did I put this on here? Okay, so this is just overview. So this is just talking about the uncoupling. That uncoupling that we were talking about is it is a release of energy as heat, because if the ATP synthase is working, you're releasing those hydrogen ions as just ATP. But now, is it because we're dissipating this proton gradient? Since we're not getting energy, it has to go somewhere, right? You know, you can't create or destroy this thing. So it has to be something. So what it is, instead of ATP, it's heat. This is one of those things that they like to associate with malignant hyperthermia in whatever the ryanodine receptor for anesthesia. But this is one of those things that can happen. So it's a release of energy at heat. It can cause a malignant hyperthermia. That's what I was trying to get. Oh, sorry guys. Uncouplers of oxidative phosphorylation. So what can actually do this? DNP, aspirin. Oh, I think I remember DNP being, I don't know. That might be something that they talk about, but just note the synthetic uncouplers, ionophores. Ionophores are anything that is going to no, I'm thinking of something else. Don't listen to me, guys. But just know what it is. Just know what it is. And then UCP1 thermogen. This is the brown adipose tissue. In infants, this is a good thing because their little bodies aren't going to hold on to heat like our adult bodies are going to. And so they have this brown adipose tissue that has thermogenin, so they can create heat so that they don't become hypothermic. So this is a good thing in infants. But as you get older, the brown fat is replaced, and you don't need it anymore. So yeah. Mitochondrial diseases, this was not a very high yield thing. I think I looked at these for like a solid 10 minutes. I had no idea what I was supposed to know about it, and I just took the L on this because it was stressing me out way too much. Just know what they are and that they exist. I don't remember if they talk about the mitochondrial diseases anywhere else in this module. If they do, know what they tell you. But like I said, I don't really remember this being wildly high yield. Gluconeogenesis. It is not the opposite of glucogenesis. Glycolysis. What did I just say? It is not the opposite of glycolysis. But there was those reversible enzymes that we were irreversible enzymes that we were talking about, the regulated enzymes. That's where you're going to have to focus on. So you can't just say, oh, go the other way. It doesn't happen like that. So there are a few things that need to happen for gluconeogenesis to take place. Purpose, you're providing glucose for the blood. So again, it's not a reverse. The biggest thing, you have to bypass this right here. And so you can't go from pyruvate to PEP. You have to do a little roundabout thing. So pyruvate carboxylase in the mitochondria, oxaloacetate, then PepCK is going to allow you to go back to PEP. Okay, so uses carbons from glucogenic amino acids, but cannot use carbons from acetyl-CoA. That's why you can't just go backwards. So acetyl-CoA cannot be used in the fall. Okay, you guys can go through that. Glucose alanine cycle. This is like a medium yield, understand this. So we're using alanine released from the muscle. So again, protein degradation, you're working out, you have a lot of muscle degradation, you need to rebuild, you need to recover. So the alanine from this can be used for gluconeogenesis. Come on. There we go. Okay, so lactate, remember Cori cycle, oxidized 2-pyruvate by lactate dehydrogenase, which can be used for gluconeogenesis, and alanine transaminated, know that enzyme, 2-pyruvate. Okay. So they talk about, I'm not going to harp on this right now because we're not talking about beta oxidation right now. They really go into this hard when you talk about beta oxidation, but just acetyl-CoA is an inhibitor to move forward. So inhibits PDH, can't move forward in it. But I'm not going to harp on that right now. But a big thing here is this reaction that have to get back to PEP. So you have the pyruvates, and then you can go to oxaloacetate, and remember that oxaloacetate can go to malate because there are a few that are reversible in this in TCA. And so malate can cross because you have that malate shuttle, or malate can cross, my God, Linz, and then can go back to oxaloacetate, which by way of PepCK will bring you to PEP. We already talked about this. So alanine, glutamine, know those two that you can use those for gluconeogenesis. This is another substrate you can use. We're not going to read that slide. And then here's another kind of detour that this has to make to do gluconeogenesis. So remember, glucose to G6P is trapping glucose in the cell because glucose is just going to go in and out because you have the glutes. And so glucose comes in, you're like, nope, we're trapping this here. We want to utilize it. So you have glucokinase or hexokinase to get G6P. But G6P, since that's the trapping of the glucose, it's not just going to go back to glucose, right? Because if you can just go back to glucose on a whim, then the glucose is going to leave the cell. So you have to do a little detour. So G6P goes into the smooth endoplasmic reticulum. It has an enzyme there, glucose 6 phosphatase, and that's how you can get your glucose back. So this is highly regulated because we don't want glucose to freely be able to leave our cells. We need glucose to do a lot of different things in our cells. So this is another detour it has to make. Beta oxidation, if you guys will remember that beta oxidation yields an insane amount of acetyl-CoA, and it can produce an insane amount of ATP. But we'll go over beta oxidation later. But fatty acid degradation provides ATP because you get the acetyl-CoAs. Provides allosteric regulators that can switch from glycolysis to gluconeogenesis. And so we'll talk about that when we get there. This is more of an introduction. Okay. Have to take place at the same time. Okay, so it's saying that both of these are taking place during fasting hepatocytes. So if you have an excess of pyruvate, you're not going to have PDH because, oh wait, never mind. Oh, all of these are inhibited. So acetyl-CoA, NADH, you have an excess of acetyl-CoA, which can come from beta oxidation. You have an in excess of NADH because the TCA is just working. It's like, no, we have enough, stop. So that's going to activate PDH kinase. Remember, PDH kinase is going to phosphorylate PDH, and phosphorylated PDH is an inactivated PDH. So pyruvate can't go through this. So it's going to go somewhere else. It's going to go gluconeogenesis, but we want that, right? We need this pyruvate to go gluconeogenesis because we have enough of everything else. You know, you don't want to go through glycolysis, TCA, if you don't need these byproducts. So it's like, nope, sorry, you can't come this way. Sorry, pyruvate, you have to go back the other way. Do you guys want a break right now? We'll take a break.
All right. Again, we're gonna try to go quick, but if there's anything y'all want us to slow down and go over, we can do that. but I don't have the chat up, so y'all just speak up if you don't want me to stop. All right. Embryology, as you know by now, is very much tested on the clinical aspects when they go wrong. Okay, so they don't ask you pathways and how everything works normally. They give you a presentation where some child has something and they ask you what was the deficit. All right, so super straightforward, foregut, midgut, hindgut. I think, yeah, this is like a good representation of it. These are easy points, right? Know what goes with what. Everything embryologically kind of falls in this nice, neat pattern here. So make sure you know where everything comes from and goes to. Including the blood vessels. I'm sure I have a slide coming up. But yeah, well, you can see here, celiac, and then superior mesenteric, inferior mesenteric. There's also those, they use those retrograde dyes, those images, those x-rays with the dye, or the CT scans with the dyes, and they ask you which one's the superior versus inferior mesenteric. Make sure you can tell the difference, because the superior mesenteric, remember, well, hopefully I have a slide coming up, but just in case I don't, remember superior mesenteric is going to go to both sides, because it'll give blood supply to that right side of the large intestine, but it also gives some blood supply to the small intestine on the left. If you look at the inferior mesenteric, it's only going to go to that distal colon. Okay, so make sure you know that. I'm pretty sure I have a slide and I'll point it out, but yeah, those are the ones they like to bring up on the exam, even that lab exam y'all are going to have at the end. Okay, right. Of course, stenosis is a narrowed lumen versus atresia is a complete blockage. They talk about child has projectile vomiting. Whether it has bile or it's bilious or not determines if it's before or after where the pancreatic duct comes out, which is right about at this the part of the duodenum, right? So it's usually superior to this major papilla. That would mean it's non-bilious, and after that, it's bilious vomiting, right? Okay, so that's one of the questions they like to ask. If it's non-bilious, sometimes, or a lot of times in the question, they'll be talking about like a pyloric stenosis or an atresia there. And of course, that's in the where the stomach joins the duodenum. So that's obviously prior to this where the pancreatic duct opens. All right. This was a little weird. Just remember this. When everything happens in embryologically in development, everything that is anterior is going to go to the right side, and everything that's posterior is going to go to the left. So it kind of shifts this way. And if you keep that in mind, you'll be able to answer these questions that talk about it, because they'll ask you where it originated from. So if you keep that in mind, you'll be able to answer just those two things. All right, that's what I mentioned: pyloric stenosis, projectile vomiting, non-bilious, right? Okay. And you can see that here. Look at this. Why do you see fluid here? Why? Okay, so they did a barium enema. They swallowed fluid. Is this atresia or stenosis? It's stenosis, right? Because even this is narrowed right here. This is where the pylorus is, or the stenosis is. But you see some fluid got through. Okay, but you can see it's narrowed right here. So and that's why the child has projectile vomiting, because it's very hard for fluid, even not just food, but fluid to get through. So that's something that's fair game. They could show you this picture. All right, right, and this is that point that where you divide the foregut to the midgut, the second part of the duodenum, right about where that papilla comes out, right? You can see the head of the pancreas right here and comes through. Questions they like to ask, they can say that there's some sort of ulceration at the first part of the duodenum. I'm sure y'all have come across this in your practice questions. They'll ask you, yeah, like there's a posterior perforation in the first part of the duodenum, and they'll ask you, this is the duodenal, pancreaduodenal, what is this one? This is hepatopanc, which one is this? Somebody tell me. Which is this vessel right here? Pancreaduodenal? Yes, no, anybody? I think that's that. That's what they'll ask you on the test. They'll say there's a posterior perforation. What is going to be injured? And it's this right at the duodenal bulb. I think it's called the hepatopanc.
The gastroduodenal.
Gastroduodenal, that's it. Thank you very much. Yes, and that's that area right there. They also like to ask the question, if there's a posterior perforation in the stomach, where does the fluid spill out to? And that is the omental bursa, or the lesser sac, right? I'm sure y'all come across that as well. So those are the questions they like to ask. But these are fair game. You know, the celiac trunk, the branches that come off. When you look at some of the imaging, you know, the spleen has to get a lot of blood. So this splenic artery that runs to the spleen is very large. So you can see that it always runs right above the pancreas. So that's kind of a landmark I like to use when I'm looking at this stuff. And then of course, this triad here that goes into the liver, that's a good landmark as well. Okay. Stenosis, atresia. So if there is atresia, you would expect polyhydramnios. Remember, the child has to be able to swallow the fluid. It helps with lung development, helps their GI tract develop. So if they can't swallow fluid, you're going to get too much amniotic fluid, right? Because it's all, the child can't swallow it. So there's too much in the surrounding. Okay, cool. Right, and then you can see this is that double bubble sign. And again, if you see this atresia there, it would be like right here. Okay, so the reason it looks like that is because the pylorus, it doesn't mean that there's a pyloric stenosis right here, but it is a valve right there, or it is so you can see that there's air in the stomach, then air gets through that sphincter, and part of the air gets here into the first part of the duodenum, but then you get full atresia, right? Air can't even get through. So you can see this is that double bubble sign. So you can see that the atresia is right here, but see this area right here correlates to this, and then this area here correlates to that. Okay, and you see this narrowing at the sphincter is right here. So the atresia is down here. Okay. All right, and they could ask you about this if the pancreas, if you have a bifid part of the ventral pancreas, if it goes over both sides, it can actually cut off the lumen here to the duodenum. So you can see it's a narrowing there, and that's what they call this annular pancreas. And so fair game, this is good buzzwords for you, a bifid ventral pancreatic. But bifid meaning two sides, so it kind of surrounds it and it kind of chokes it off right there. All right, not that these aren't super important. I don't think they asked us about this. Some of these rotation problems, maybe just a simple definition for each will be fine. Umbilical hernia, fairly common. Good thing to know why it happens. All right, so they can give you a presentation, not tell you what it is, but give you the presentation for it, and they'll say what happened, right? Didn't close, incomplete closure of the ring. Omphalocele, I remember seal means it's sealed off, right? So you do get a membrane around it, okay? Whereas gastroschisis does not have a membrane around it, okay? So that would make sense that they would be prone to hyperthermia. So they could ask you these, if they're asking you to differentiate these, one has a membrane, one does not. Buzzwords here. For gastroschisis, you don't get those lateral folds closing over. So you get no membrane, nothing covering that. All right, Meckel's diverticulum, remember the rule of twos, 2% of the population, men 2 inches long. What's the other one? Two types of ectopic tissue, usually gastric and pancreatic. That's a good thing to know there. I think it's 2 feet from the ileocecal valve. There it is, yeah. And then, yeah. So usually they find this incidentally. Sometimes they're older, but it can mimic signs of appendicitis, that early on appendicitis where you get this, midline pain right here. So they'll bring the patient in thinking they have appendicitis, and then they'll find this. All right, and this is from first aid. See that here, it's a little diverticulum. Yeah. Oh, it even says right lower quadrant pain, so it could actually present even further along with appendicitis. Yeah, okay. Cyst, right? A cyst formation. Remember, a fistula has an opening, so you can get fecal matter coming through the fistula there. And anal canal, yeah. Just remember when they talk about, I think y'all talked about this already, but when they do, what do you call it? Hemorrhoids, internal hemorrhoids are painless versus external are painful because of their innervation, right? The external ones tend to have, are somatically innervated, so they're more painful. For all these little variations here, just knowing what's in red, these little differentials, then you should be fine. I think we might have had one question on it. So just know the differences there and like what happens to create it. Yeah, I wouldn't call this high yield at all. Hirschsprungs, yes, congenital megacolon, that is important. They'll say the child is not able to pass meconium, which is that first defecation. So you get this this megacolon here. So what happens is you don't get any innervation, so you don't get peristalsis at this area down here. So you get this backlog of fecal matter. So what's very important, as always, failure of migration of the neurocrest cells. There you go, easy points. All right, so these neurocrest cells didn't get down here, and so they so there's no peristalsis down here. All right, autonomics. So much of this is review for you guys. It's one of those things you just have to suffer through, but it's so important, especially as you get to second year. Lots of drug targets here. You can see foregut, midgut, hindgut, you know, the innervations there. So the parasympathetics are craniosacral, remember the vagus nerve, and then the hindgut has the pelvic splanchnics, P pelvic, parasympathetic. And then the, well, let's see. So the sympathetics are going to come from this thoracolumbar chain here, the sympathetic chain that comes through here. So be sure you know that. Oh, and innervation. So we're talking about digestion here. So we're thinking parasympathetics. But remember, the foregut and the midgut is supplied by the vagus nerve, the cranial portion of the vagus nerve, and the hindgut comes from these pelvic splanchnics. Okay. Now, again, typically talking about parasympathetics, GI motility, you know, rest and digest, whereas sympathetics, you know, vasoconstriction, constriction of sphincters. You don't want to go to the bathroom when you're running from a bear, right? The enteric system can run independently, but it does have parasympathetic components. It actually has sympathetic and parasympathetic components, but the way we talk about it, it's generally parasympathetic, okay? Because we're talking about that, that motility. Right, so this breaks down very simply. Everything goes together. There's no crossover. So greater splanchnics for the foregut, lesser least midgut, and then hindgut is lumbar splanchnics. Okay, so good points, easy points there. Pelvic pain line, y'all have learned this. Y'all did reproduction, but when we talk about in the gut, we talk that pain line is the midpoint of the sigmoid colon. Okay, so where we talked about it in reproduction, it was that the bottom part of the cervix. When we talk about it here, it's the midpoint of the sigmoid colon. All right, again, they like testing on this, these structures, how everything breaks down embryologically, but you should know where the referred pain goes to. Yeah, midgut would be periumbilical. It falls in line, right? You expect foregut to be epigastric, midgut periumbilical, and then hindgut goes down to the flank or groin pain. And then once you get after this pelvic pain line, it's perineal pain, okay? So make sure you know that this pelvic pain line, that's where the switch off is from sympathetics to parasympathetics. So that just means that those visceral pain fibers, those visceral afferents above in the hindgut, above that midpoint of the sigmoid colon, they travel with the sympathetics. Below that point, all those visceral afferent fibers will travel with the parasympathetics. Okay. Appendicitis, when you patient first gets appendicitis, they'll talk about this. It's a referred pain. It's this periumbilical pain that you'd feel here. Visceral pain, very dull. But once it gets bad enough, it can actually irritate the parietal peritoneal membrane, parietal peritoneum. Sorry. Yeah, that's right. And then remember that the parietal peritoneum is innervated somatically. So then you can actually pinpoint this pain. So it has to get really bad before they actually get it in this lower quadrant there, that McBurney's point, that you feel it. Okay, and remember, whenever they talk about rebound tenderness, that means the pain is felt when you lift off. So what you're actually doing is pressing the appendix, that inflamed appendix up against that parietal peritoneum. That's what you're doing with the referred pain. And when you lift off like that, that's when the patient feels the pain, okay? Gallbladder pain is typically felt, you can see here, this second area, T7 through T9. So if they ever say the patient feels mid-back pain on the right side, that typically is some sort of gallbladder pain. It could be due to some sort of stones or something like that. Shoulder pain typically comes from the diaphragm, that right shoulder. This is the slide I was talking about. Of course, you definitely need to know this, know where everything falls, know the sympathetics, know where the parasympathetics go, know the arteries that are involved. All right, anatomy. So, right. Know this. Intraperitoneal versus retro. Yeah, I mean, like, it's just which ones fall in the peritoneum, which ones are behind, or the kidneys fall behind. And then some are primary versus secondary. I think there's here it is. Yeah, memorize these so you know what goes with what. There was one, oh yeah. So like the duodenum is split. The first part will go through the intraperitoneal. What else is split? Oh yeah, the transverse and sigmoid colon are split too. Because the ascending and descending actually travel in the back. What you should do is go look at first aid. Did I put first aid slide? I did. Here we go. Okay, hold on. So then when we talk about secondary retroperitoneal, we look here, you can see how the pancreas kind of fits in the middle. So it happens during development. They get this mesentery forms around them. Okay, so this is secondarily retroperitoneal. I don't know that they'll go that in detail with it. They'll just ask you which one's intra versus retro. And this is the slide I wanted to point out. Sad pucker. So this is the ones that fall retroperitoneal, right? And so some of the tricks, right? So second through fourth of the duodenum. So the first part is intra, and then it dives back, and then colon, descending and ascending are in the back. So don't get those confused. All right, and then the cavity. Remember, I mentioned that those posterior perforations of the stomach will fall into the lesser sac. That's a good question they like to ask. Same thing here. Right, hepatoduodenal ligament. This is a lot of memorization. Yeah, this Pringle maneuver, they use that, the hepatoduodenal ligament, to prevent blood into the liver. So you can stop bleeding there for traumatic injuries. Okay, so they could ask you about that and ask you what's the components of this. When they press on the liver, remember that triad's down there as well. So that's actually what they're pressing on. All right, more of the same stuff. Paracolic gutters, remember those? There was a difference. Yeah, here you go. Left one is narrower. So if it's gonna go down a side, it would preferentially go down the right side. Easier to go down the right, yeah. Very important when you have some sort of trauma, you're worried about internal bleeding, the pouch of Morrison. If the patient is laying supine, that's where blood will collect. If the patient is standing up, it'll be in the pouch of Douglas. All right, and you can see that here, or see it here, this pouch of Douglas, and then in the man, the retrovesicular pouch, okay? Easy points. Now, there's constrictions of the esophagus. They usually talk about 3 points, but this has 4. So you have the junction, or the upper esophageal sphincter and the lower esophageal sphincter. And then you also have where it crosses the aorta. These are kind of close together. So and it's compressed by the bronchus as well. So they kind of go together here, whether you want to call that one constriction or 2. I guess it's up to you. But keep in mind also that the first part of the esophagus is skeletal muscle, so it's voluntarily controlled. Lower is smooth, and mixes in the middle. Gastroduodenal, there it is, yes, right? So that's the one that falls below. They said there's a posterior preparation to the duodenal bulb. That's what's going to be affected, that gastroduodenal. But again, this is just memorization from first aid. All right, there we go. That perforated ulcer, right? Also, this what's this called? This air pocket is like a hemiperitoneum or something like that. That's where the air is going to develop. If you have some sort of perforation, that's the highest point. Again, yeah, there you go. That's what I had mentioned earlier. All right, let's see. Right, be able to differentiate these on the enemas, right? On these barium enemas, you can see that the jejunum has this feathery appearance, where the ileum is more smooth, and of course the Paris patches, that's the immune system of the gut. These are lymphoid tissues or nodules that are there that you could find in the ileum. All right, and now this is what I wish I had the picture. I guess I don't. Okay, so you know that the superior mesenteric comes off. Remember, I said that it goes to both sides, right? So it's going to give the ascending colon blood supply, but it also comes off to the small intestine over here. So if they give you some sort of CT scan with some dye, be able to differentiate that. The inferior mesenteric is only going to go to the patient's left side. Okay, in case they ask, I don't know that they will. You can ask about this, the jejunum, long vasa recta versus the ileum. It's just another differential there. All right, we talked about the appendix. You can see the small bowel obstruction. Obviously, you would expect nausea, vomiting, stuff like that, and you get this stack of coins appearance, which is more of this plica semilunaris, right? So that's part of the luminal wall of the small intestine. Okay. And you can kind of see this stacking here. Right, and then we get to the large intestine, some of the differential here, this Taenia coli, they like the haustra. You can really see that when they use those dyes. You can see these separations here in the haustra. So be able to note that, differentiate that. We talked about this, everything below the pectinate line, those external hemorrhoids, somatically innervated, those are painful. Endodermal or visceral afferents above it, those are painless hemorrhoids. So that's the medical or the clinical aspect they like to use when they teach that or when they test on that. Right, so you really do get dual blood supply. It's sort of like the uterus, right? You get some blood supply from the top, some from below. I don't know that this is super high yield. Same thing here, the hemorrhoids, that's what they like to test on. All right, so they love to test on the anastomoses. With the prevalence of drinking and alcoholism, among other things, hepatitis as well, patients tend to get liver cirrhosis. With cirrhosis, you get portal hypertension. Why do you get that? Because the blood cannot flow properly into a cirrhotic liver, so you get this portal hypertension. So what happens to the blood? It has to go somewhere else. So that's where you develop these anastomoses. So I guess I have it somewhere else. We'll go through it in a second. But yeah, that's the idea there. So there's a couple you need to know because they like to test on it where there's the esophagus or in the rectum, but we'll get there in a second. Diverticulosis, you can see this. Remember, it's painless rectal bleeding. It's above that pectinate line or the pelvic pain line. So it's painless. Diverticulosis is just these outpouchings. If they get infected, it becomes diverticulitis, right? So this is fairly common, but it's painless. So patients don't usually complain until it gets really bad. Sigmoid volvulus just means it's spun around. So you get, not only do you get a cutoff of blood supply and ischemia, but the fecal matter can't go either. So this is obviously a very extreme situation. It has to be corrected quickly. You can see that here, look at that, yeah. All right, it's good. So here's a good example of it. So if they give you this, they would probably give it to you a picture of that dye, that retrograde dye that they use, but you could see that some of the blood supply is gonna go to the, to the right colon, but the superior mesenteric, it's tricky here because this is just showing the large intestine, but the superior mesenteric also gives blood supply on this side to the small intestine. That's not shown here. But this inferior mesenteric is only going to go to this colon. One thing they do like to test on are these marginal arteries. So this is the splenic flexure over here. As you can see, it doesn't really get a good blood supply. You don't get these direct branches. So the only way you can get blood there is through these marginal arteries. So sometimes they'll tell you, there's an ischemic area, or they'll tell you about the splenic flexure and they'll ask you, how does it get blood supply? So it's kind of like a side road, right? So you have to get it through there. So watershed area is the word that they like to use for that. It just doesn't get a lot of blood. Okay. And then the venous system is very similar to the arterial system. One thing, I think it came up on the lab exam, just take note, the inferior mesenteric joins the splenic, and then the superior mesenteric comes in here. I think it was on the lab exam, so you don't need to worry about it right now. But if you look at this on one of those dye images, sometimes it's confusing because they'll point to this one and they'll be like, is that superior or inferior? Just know that the inferior joins the splenic and then they come together and they join the superior. So yeah. And then, yeah, the lymphatic system.
Sorry, I know you said you
Yes.
were looking at the chat, so I had a question.
Yeah, sure.
The marginal artery, that one is the one that supplies the watershed area. Is that correct?
Right, yeah, it's like a back road or like a side road, right, to the interstate. So like if these are big branches here, this marginal artery just kind of runs around and kind of gives blood to the areas. But yeah, specifically, like if the marginal artery was affected somehow, the splenic flexure would be the first area that would be affected because it's dependent on it. Yeah.
Okay, thanks.
No problem. All right. Porta hepatis has all of these: the duct, hepatic artery, and the portal vein. That's that triad that we talked about. Let's see. Right. Again, this is just straight anatomy, so you should know the different parts of the biliary apparatus or the duct system. Now, depending on where the blockage is, you can tell what, the lab values will tell you where the blockage is. For example, if you were just blocking the liver up here, you get an elevated AST and ALT. If you were just blocking the gallbladder over here, you get an elevated GGT and ALP. Not going far, yeah. But if you're blocked down here, meaning you're blocking the gallbladder and the hepatic ducts, then you'll get elevation of all four: AST, ALT, GGT, and ALP, right? So the lab values will tell you where the blockage is. And if you get a blockage down here, you can block the pancreatic duct as well. And you get an elevated amylase and lipase. Okay, so make sure you know that. All right, this is just from first aid, same thing. And big buzzword, after a fatty meal, they get right upper quadrant pain. And that's because the bile helps to digest fat. So when you get a fatty meal, it's supposed to be contracted. That CCK contracts the bile duct. And if you have stones there, then you get pain. Okay. Endoscopic retrograde. So you can see this tube here. They went into the port right where the duodenum, that little opening, the papilla, and then they shot dye up it so they could see the pancreatic duct here, and this is the common bile duct. This is where it branches off to the gallbladder, and then the right and left hepatic ducts. Okay, so you should be able to recognize that. I don't know if that's super important. Let's see. Yeah, again, I mentioned that splenic artery is very large and it runs on top of the pancreas, so be aware of that. Portal vein, is this where we talk about it? Yeah, so this is super important. So these are these anastomoses. When you have portal hypertension, the blood has to go somewhere. So you end up making these anastomoses, and it's just this backflow, which causes problem because these are very fragile, right? So when patient comes in and they're coughing up blood, hematemesis, something like that. Yeah, am I saying that wrong? Hematemesis, it sounds weird. Sorry. Yeah, so they're coughing up blood or they're throwing up blood because these rupture of these veins down here. And same thing if they're defecating blood, it could be at in the rectum. So just make sure you know what the anastomosis is. So the purple with the blue here. That's how it forms. Umbilicus as well. That's where you get the caput medusa, And then 4 and 5 aren't super important. They don't ask about these, but those 3 are definitely important. You need to know where the dilation is and where the problem is. Okay. So yeah, more anatomy, pretty straightforward. Let's see. Clinical correlations, yeah. So any sort of tumor or mass in the head of the pancreas can cause problems. That's typically where these tumors form. So it can cause a problem with this outflow tract or even compress the duodenum. All right, so yeah, you should be able to look at these. This will probably be more for the lab exam, but yeah, you could see these coming off the common and the proper, and then the gastroduodenal coming down, and then the spleen over here, the splenic, right? Yeah. Barium swallow. So we're looking at the stomach, pyloric sphincter, the duodenum first and second part. Right, definitely, of course, the jejunum has the feathery appearance versus the ileum. Large intestine, remember we talked about those hostra, look at these separations here. So you'll be able to identify that. Here we go. So look, so superior mesenteric, these branches 1 and 2 are coming off to the right colon, okay, the ascending colon. 4 and 5 are giving branches off to this small intestine on this side. So make sure you can tell the difference because they look kind of similar, superior versus inferior. So if you're looking at inferior over here, remember it's only going to go to the left colon, okay, the descending colon. So make sure you can tell the difference between these 2. All right, yeah, pretty straightforward. I don't know that they'll really ask you any of these. You can see that large splenic artery come across right above the pancreas here, large aorta right here. The dark shaded area is the gallbladder. Right, okay, so this is that picture I was telling you about, right? So they'll ask, they would like point to this right here and you'll be like, is that the inferior or is it the superior? So just remember this inferior, which kind of dives down, you can't see it, joins the splenic, and then the inferior and splenic come together to join the superior mesenteric, okay? So we did have a picture like this and they were pointing to this one and we were like, I don't know. So that's how that works. It has to join and then it makes the portal vein, right? That goes directly into the liver. All right, glycogen. We kind of talked about this already. So I'm going to not go over it for the sake of time. Yeah, I'll just go over some brief points. You should know the difference between hexokinase and glucokinase, specifically in regards to Km versus Vmax. Okay, so you want to have a lower Km, which is a high affinity for glucose in the muscle, so it uses it faster. And Vmax correlates to being able to store a lot. So you want to be able to store a lot or have a high capacity in the liver for glucose. Okay, so Km is high, or sorry, Km is low. They're inversely proportional. So Km is low, meaning a high affinity in the muscle with hexokinase. Glucokinase has a high Vmax, so it has a high storage capacity. Make sure you know that the feedback inhibition is different as well, okay? We talked about this, bifunctional. If y'all see anything y'all want me to talk about, just mention it, but we kind of talked about these already. Okay. Right, so this should make sense. If you're trying to run away from whatever that is, an elephant, I suppose. Epinephrine short term can mobilize, and so it could help with glycogen degradation, right? So you can mobilize energy in the muscle, okay? So that should make sense to you. So we're going to phosphorylate these kinases. Remember, by phosphorylating, we are increasing blood glucose, right? We're increasing the breakdown of glycogen to upregulate or to make more glucose. That way the muscle can use it in this instance. All right, yeah, same thing here. So you can see that once you mobilize it, again, phosphorylating it. Remember, dephosphorylation is the insulin pathway for storage. Phosphorylating is the glucagon pathway for using energy or making glucose so that you can use energy. All right. know this one. MODY2 is kind of like it's similar to diabetes, right? But it's specifically a glucokinase deficiency. So easy points. Make sure you know that that's what that correlates to. So again, where would this be, right? It would not be in the muscle. You wouldn't be worried about this in the muscle. This is specifically in the liver. So these kids will have liver problems or difficulty mobilizing glucose from the liver. Right. I want to get to more of the clinical aspects. That's what's important. The patient's not going to come in and ask you what the rate limiting enzyme is. So yeah, they're going to ask you about clinical aspects. Same stuff here. We talked about this again, dephosphorylation versus phosphorylation. Right, remember that, I think I had this wrong in my head. So the 1,4, somebody, is that 1,4s are the linear change and the 1,6s are the branch points? I think I had that backwards in my head, but yeah, okay, that's what this implies here. So when we talk about those glycogen storage diseases, we'll talk about that. But you can imagine these large glycogen molecules have a ton of glucose molecules connected to it. So if they were just in a single chain, they'd be really big. So that's why you put branch points here to kind of make them smaller. So there are complications with that with the glycogen storage diseases as well. And here they are. So definitely note these kind of backwards and forwards. Actually, you need to know what enzyme is involved, and you know what the problem is. Did I go through them? Oh, so I have them in first aid. Another big thing they like to ask is, they'll say in the stem of the question, they'll be like, does the glycogen molecule look normal, right? So if it does, so if it doesn't look normal, that's usually a problem with one of these two, right? Because you have a problem with the branch points, the debranching and the branching enzymes. So they won't look normal, but some of these other ones, they'll look normal. So just take note of that, because that's a giveaway for what's going on. And if you have Anderson disease, you have a problem branching it. So you have a problem making the proper glycogen molecule. If you have problem with Corey disease, you have a problem debranching it. So you have a problem breaking down that glycogen molecule. Okay, so keep that in mind. McArdle's is specifically for the muscle. I think we'll go through it in a little bit more detail, but here it is in words from first aid. What was the one, because they do the thing, is it painful muscle cramps with strenuous exercise? Isn't it like lactate doesn't go up? I think that's what it is. They'll throw that in the stem of the question too. They'll say they were working out and the lactate didn't go up, which it should, right? If you have strenuous exercise. So that's a giveaway for McArdle's. All right, cool. So we'll go through them. Remember, von Gerich's is very, very severe. So you have a normal structure, so you actually have the actual glycogen molecules, but you can't break it down. Importantly, not only do you have storage of glycogen in the liver, but you also have it in the kidney cortex, right? I remember Dr. Trotz making a big deal out of that, but they'll tell you the kid comes in and they have liver and kidney problems, all right? So, and it's very severe, and the normal structure, the glycogen looks normal. So that's von Gerich's, okay? So very severe hyperglycemia. Why is that? Because when they're hypoglycemic, you want to be able to take the glycogen and make glucose, and they can't. So they have to have frequent meals. They'll develop lactic acidosis because there's no glucose there, right? So that's the only process you can go through, this anaerobic process. Yeah. So just keep in mind liver and renal problems, von Gerich. Pompe sounds like a pump. Right, so it's a problem with the lysosomes, right? The lysosomal glycogen, specifically this 1,4-glucosidase. But when they talk about pump, these kids typically have heart problems. Okay, so Pompe sounds like pump. Cardiomegaly, there you go, and also hepatomegaly. All right, so now this is what I was saying. So with Corey disease, abnormal glycogen. Why is it abnormal? Because you can't debranch it. Okay, so you get to the point where you get to this limit dextrin, where you just have one glucose on this little branch point, this 1,6 branch point, and at that point they can't do anything about it. Like you can't take this little this little single glucose off of there. So so that's why it looks abnormal. And so that's the problem with this debranching enzyme. You can't properly get rid of it. It's usually like 1 to 4, I guess, 1 to 3. And that's what they call, that is the limit dextrin. So that's why this is all called limit dextrinosis, because you can break down the glycogen up to a certain point, okay? But you actually need a different enzyme to take these little branch points off. So if you don't have that, you get these abnormal structures. Now, obviously it's milder than von Gehrig's, right? Because you can break it down to a certain degree. You just get these weird little, these little limit dextrins that are sticking out. So it's not as bad as like von Gehrig's, where you can't break it down at all. Again, now when we look at Anderson, same thing, but you're not able to branch it, okay? So you need to know that. So again, these are gonna look abnormal as well, right? You're gonna have these long chains. Why? Because you can't properly branch it. You can't properly form the glycogen. All right, infantile cirrhosis, that's a good buzzword for this as well. Again, abnormal glycogen structure. McArdle is in the muscle, rhabdomyolysis, skeletal muscle breakdown, okay? So typically rhabdomyolysis, if it's bad enough, can lead to kidney issues, but that's what's happening when these kids or patients start working out. That skeletal muscle will break down. They don't have ATP there. CKMM is increased, M for the muscle, right? CKMB is for the heart, MM's for the muscle. This is what I was talking about. This is the giveaway that they like to use. Patient was working out and their blood lactate does not go up. You would expect that to happen, but because of the enzymatic problem here, you can see here that lactate never goes up. Okay, so that's diagnostic for McArdle's disease. Hers is like McArdle's, but for the liver. Okay, so it's the same thing, but it's just instead of where McArdle's is in the liver, hers is a hepatic problem. So you will get normal glycogen. It's not a problem with the branching points, but it's hepatic phosphorylase versus this muscle phosphorylase. Okay. Terui's disease is just like McArdle's, but if they talk about hemolysis, okay, then you're actually talking about Terui's disease. So yeah, so keep in mind, don't get rhabdomyolysis with McArdle's, that's skeletal muscle breakdown, confused with hemolysis, because Terui's is going to get actually lysis of the blood cells. So there's PFK1, remember the red blood cells are completely dependent on glycolysis. They don't have mitochondria. So if you stress them out too much, they'll just rupture, patient comes in, bloody urine, stuff like that, okay? This is busy for me. I skip over stuff like this. But if you like this stuff, go for it. It has everything together. All right, fructose and galactose. Right, so when is fructose added to the diet? These are things they put in the question. Kid diagnosed, I think 6 months, 6 or 9 months, they start adding fruits to the child's diet. And that's when this develops this fructose problem. So that's a giveaway for it. Yeah, dependent on how bad it is. Yeah, so the fructokinase deficiency, not as bad as this aldolase B deficiency, but they go into it. So essential fructosuria is the fructokinase, typically asymptomatic. It's benign, whereas aldolase B deficiency, this is much worse. Here we go. After weaning and fruits added 6 to 8 months of life. So giveaway, right? Whereas the galactose, we'll see in a second, usually it happens from birth, from breast milk or from milk, but they usually become symptomatic at like 2 weeks, I think it is. They start getting cataracts that early, in only a 2-week period. But I'll show you that in a second. So again, we're looking at timeframes here to be able to separate these. Yeah, big point here. When we talk about galactose deficiencies, fructose they're not going to get the cataracts that form, but you will get hepatocellular failure and jaundice because of this. And look at this, hypoglycemia following fructose in the diet. And you'll find the fructose in the urine because they can't break it down. Again, dead giveaway, 6 to 8 months. All right, this is just from first aid, kind of outlines it. Remember the aldolase B one is worse. All right, so now what are we doing? Oh, right. So diabetes. So what you get in in diabetes, the cataracts that form are from sorbitol buildup. You do have aldose reductase in the eye, so you can make sorbitol. For people that don't have elevated glucose, it's not a big deal, but for people that have diabetes and their sugar's like 400, you get a ton of sorbitol that builds up. So if that's the case, the eyes don't have sorbitol dehydrogenase to break it down, so you get this sorbitol buildup. So they can ask you that to differentiate the cataracts in diabetes versus the galactosemias. So just remember, with the cataracts here with diabetes, it's a sorbitol buildup. But honestly, like these patients will be older versus the ones in galactosemia, right? But again, so now the cataracts that form here are galactitol. That's what forms there. But take note, aldose reductase is used in both situations. Okay, it's the enzyme that forms galactitol and sorbitol. Now, galactokinase deficiency, it's bad, but not as bad. This is a longer enzyme. The longer the enzyme, the worse it is typically. Right, so the classical galactosemia, that's the one that's really bad. So look, the galactitol that's built up, again, these will be in kids, whereas the diabetes will tend to be in older patients. But you can see this galactitol that forms in the eyes. So again, they start drinking breast milk or milk in general, and it takes about 2 weeks before it starts being noticeable. They also get intellectual disability, so this is a big deal. It's better to be diagnosed prior to you get to the point where these cataracts start forming, because that's the point at which, you know, there's actually damage that's being done. So again, here you go, 2 to 3 weeks of life. Whereas with the fructose problem, we're looking at about 6 to 8 months. Okay, so it's not saying that this is when it started. It started from birth when they started ingesting the milk, but it starts to present in just 2 to 3 weeks. Okay, that galactitol starts to build up, jaundice, hepatomegaly, hypoglycemia as well. So keep in mind that. Right. Of course, you'll see the galactose in their urine because they can't break it down. And again, neurologic development. So at 2 weeks already, you get cataracts. You need to stop them and limit that from their diet to prevent further developmental delays. This is from first aid. And that's a good giveaway here. They might not tell you what the reducing sugar because it gives it away right away, but they may just say there is a reducing sugar in the urine, which means by definition, they can't break it down. It's supposed to be able to be reduced and it can't. All right, lactose. Usually they talk about lactose deficiency in regards to the brush border, because that's where you get that lactase. If there's some sort of infection, it could be, you can have secondary lactose deficiency there. So there are different versions of it. Just knowing the enzyme or the genes involved here, you should be fine. Yeah. Yeah, of course you get osmotic diarrhea with that. Like severe, right? So like where you get like, you'd have to be hospitalized for that sort of dehydration at that point. So again, we're looking at that lactase in the brush border. And then secondary, right? Usually it's some sort of infection. Rotavirus is a big one in kids. So they get this intestinal injury, breaks down the brush border. You don't have the lactase, then they get this osmotic diarrhea that's formed. Okay, so just be aware of that. And again, this is from first aid. All right, fatty acids, right? So the basis of fatty acids are tags, right? You triacylglycerides, you take these fatty acid chains, you kind of hook them up, and you hook them up to this glycerol, and you have tags. Now the tags float around, and they end up making, you start making larger fatty acids, and well, we'll get there in a second, right? So, right, of course, once after you eat, you want to store all of your glucose. One of the best ways to store glucose is making fatty acids, right? And like, if this is confusing, like I know there's a lot of steps, and honestly, they could spend 6 months explaining like just fatty acids. It's very complicated, this whole process. So just keep in mind the very important enzymes, those rate limiting enzymes that are necessary for it, because like I said earlier, in the question, they'll just say the patient hasn't eaten in 2 days and like, what's going to happen? What's going to happen? And then they'll give you a list of enzymes. So you just need to be like, well, they're going to break down fats, beta oxidation, right? So you're going to break it down, and you'll just need to know the rate limiting enzyme for beta oxidation. Okay, so don't make it more complicated than it needs to be. Remember, acetyl-CoA is just a 2-carbon molecule, and that's going to be our baseline for how we're going to be making these larger fatty acids. Okay. Here's a good one. Once you get acetyl-CoA, you hook another carbon up to it. So malonyl-CoA is a 3-carbon chain, but the big enzyme you're going to want to remember is acetyl-CoA carboxylase. So if they tell you the patient just ate and they're going to store fats and they give you a list of enzymes, acetyl-CoA carboxylase is going to be the one that's going to drive fatty acid synthesis. Okay, and it gets to this palmitate, which is a 16-carbon carbon chain, and then it breaks down or goes into this fatty acyl-CoA and stuff like that. And then you end up getting the VLDLs and right, and then LDLs and all that. So again, when they talk about fatty acid synthesis, the first thing you should think of is this acetyl-CoA carboxylase. All right, now, should it make sense that it's dephosphorylated when it's active? Yes, right? Making fatty acids or making these fat chains is going to be a process of storage, right? Insulin, high blood glucose, dephosphorylation, we're storing stuff. Okay, so it should be dephosphorylated. That should make sense. Of course, if it's inactivated, it would be phosphorylated, right? So it's on insulin's team, right? We're storing stuff. Yeah, this is further on in the process, making the palmitate, right? To get it up, you're just adding acetyl-CoA. You just add 2 carbons on again and again with this until you get to palmitate. I don't know what they'll ask you about this. Right, omega-6. Remember the trick, linoleic acid, the 3, that means the N is the third to last. 1, 2, 3 for omega-3. And linoleic acid, the 6, that means the N, 1, 2, 3, 4, 5, 6. That's how I remember these. All right, so it's where the N falls. That's versus the omega-3 versus omega-6s. Okay, but you need to get these in your diet. They are required essential fatty acids. All right, again, acetyl-CoA carboxylase. Should make sense that fatty acyl-CoA, this is a downstream product of it, okay? So if you have a lot of that, that means you've been making a lot of fatty acids. So now you can tell that rate-limiting enzyme back in the front, this acetyl-CoA carboxylase, that it could be turned off because we have a lot of fatty acids being formed already, okay? Now, beta oxidation is the process of breaking it down. So when you get to this, you should think of the enzymes required for it. So fatty acyl-CoA synthetase, you see that here. This is the process of activation of breaking it down, right? And this is that carnitine shuttle. This is a bit confusing. But again, you're starting out with this fatty acyl-CoA synthetase, and you're going to go through the process of breaking down these, or beta oxidation of breaking down these larger fatty acids. Okay. And you can see that here, just the process, you just knock off 2 fatty acids that are 2 carbons of this fatty acid at a time, and you get a bunch of acetyl-CoA. And then from there, you can take acetyl-CoA, put it right into the Krebs cycle and start making energy. All right. Yeah. Why is this important? Inhibit CPT1. Oh, okay. So once you get to a point where you have this 3-carbon, you can actually tell it to stop breaking stuff down, because you have, actually, this is important. Sorry. Okay. So yeah, they'll ask you this. They'll say, what is the product that helps to stop beta oxidation? So this is the point where you're breaking down a lot of fatty acids if you're going to get to malonyl-CoA. So if you get a lot of that, you're not going to want to keep breaking down more fatty acids. You're going to want to stop beta oxidation. So if you get a lot of malonyl-CoA, it'll stop it. Okay. And if you want to go through this, you can. I don't know that they're going to give you any math like that. Okay. The big one here is that MCAD deficiency falls in this 10 chain or 10 carbon fatty acid. So we'll talk about that in a second. Oh, here it is. Remember, they're going to give you in the question hypoglycemia and hypoketosis, right? You can't make glucose and you can't make ketones. You're not able to break down the fatty acids. So if that's the case, they're going to be talking about MCAD deficiency. So they'll put that in the question. The patient has low glucose and low ketones. Right away, you know it's MCAD deficiency. And yeah, this is the product. This is diagnostic. If you get dicarboxylic acid in the urine, that means that it's diagnostic for MCAD deficiency, and you should expect to see this through hypoglycemia and hypoketonemia. And then there's some other deficiencies here, carnitine deficiency as well, whether it's systemic or in the liver versus myopathic, just a little different presentations. And these fall in that category too. This one systemic is in the liver, whereas CPT2 is myopathic. Okay, so just keep those in mind. Same thing, right? So we're looking at liver versus in the muscle. So just keep those separated in your head because they kind of go together. MCAD and CPT1 versus the myopathic carnitine and CPT2, liver versus muscle. This is the process of actually breaking down or the MCAD, or sorry, inhibiting the MCAD process. So this Jamaican vomiting sickness has an enzyme that helps, that will induce MCAD deficiency, just in case they ask you about that. This isn't super important. Remember, carboxylases use biotin, but if you do get to a point where it's odd chain, how do you get acetyl-CoAs? There's 2 of them. So if you get an odd chain, you have to do something about it. And this is that process of breaking down 3 carbon chains. Let's see. Right, so low insulin glucagon ratio. Okay, so in a fasting state will induce hormone sensitive lipase. So that will help you to break down lipids. So what team is hormone sensitive lipase on? It's on glucagon's team. Hormone sensitive lipase will help to break down lipids, lipase, lipid enzyme, break down lipids, right? So whereas, what's the other one? Sorry, I can't remember. There's another lipase that's actually on lipoprotein.
Lipoprotein.
Yeah, that's it. Thank you very much. Lipoprotein lipase is on insulin's team. Okay, so lipoprotein lipase will be elevated when insulin's around, whereas hormone sensitive lipase will help to break down fats with glucagon. And then this is just from first aid if you want to go ahead and read that. All right, ketones. Zellweger syndrome, that happens to be a problem with the peroxisomes, as you can see there. So it's a problem with breaking down these very long chain fatty acids, and this is kind of how they present neurological manifestations, demyelination, hepatomegaly, hepatocellular failure. Okay, so usually they don't make it past infancy. And then Ruffsome disease is like a milder form of that. It's also in the peroxisomes. Some buzzwords for you: alpha oxidation is the process that's affected in Ruffsome disease. Okay, this phytic acid is another buzzword for you as well. All right, so acetyl-CoA, of course, once we're breaking down those fatty acids, we have tons of acetyl-CoA floating around. So it could go through the Krebs cycle, it could go to ketogenesis, make ketone bodies, beta oxidation, like we said, that was the initial process of breaking it down, okay? So the big ones you need to know, the main one is beta hydroxybutyrate, acetoacetate 2, and acetone is involved. Those are the 3 ketone bodies you need to be aware of. So if these ever show up in the urine, you know, typically when patients diabetic ketoacidosis, they'll show up with this in the urine, and you know they're in the process of making ketones. So that question we had earlier, how do you know the difference between DKA versus ketosis and starvation? And the answer is that you look at the glucose levels, because patients that are in diabetic ketoacidosis are going to have very high levels of glucose. And they're just not able to break it down. So even it's like, I think Dr. Impadi put it, it's like a disease in the presence of excess. Like you have all this glucose around, you just can't do anything with it, right? Because you don't have the insulin. So they end up making ketones, even though all the glucose is there. Whereas ketosis and starvation, obviously the patient's going to bottom out. They're not going to have a lot of glucose around. So they're inevitably going to end up making ketones. Okay. So again, yep.
Sorry, I have a quick question. So for ketogenesis, like the normal pathway, we're going to be expecting the 2-acetyl-CoA to be coming from fatty acid oxidation, not from like the pyruvate 2-acetyl-CoA, right?
Right. And because if you were going through the pyruvate Krebs cycle, you're not worried about ketones. Like you have glucose, so like you're good, right? So like you're not worried about it. Ketosis is like we have no glucose around, like we have to break down all this fat. Like we don't have another option. So then you go through this process, yeah. So again, this is very complicated of a process, but what you need to keep in mind when you think of ketogenesis is this enzyme right here, mitochondrial HMG-CoA synthase. That is going to be the main one involved with ketogenesis. So you can see here, you're first going to make acetoacetate, and then primarily you're going to make 3-hydroxybutyrate. All right, now, where do you need it? Ideally, the brain's going to use all the glucose it can, but if you're in ketosis, these are the muscles that are going to need it primarily just so you can survive. Okay, so it's not an ideal process. It's kind of, obviously it's you're in a process of starvation. Now, if you were to end up breaking down the ketones, so whereas we said making ketones, you use mitochondrial HMG-CoA synthase. If you're going to break down these ketones, this is the enzyme you need to keep in mind. Know both names for it. Succinyl-CoA acetoacetate, CoA transferase, or thiophorase. Make sure you know that it is only present in the peripheral tissue because those are the muscles that need it, the skeletal muscle, the heart, and the brain, right? So that's where you're going to be able to break down these ketones and then get it into the Krebs cycle, okay? Right. So, did I put it? Okay, so yeah, so you just see the process of breaking it down, beta oxidation. So this is the question that was just asked. You'd see the beta oxidation. Okay, so we get all these acetyl-CoAs. Now, one of the problems here is to go through this Krebs cycle, you need to have NAD, right, to be able to break down to make NADH. So if you're in the case where you get increased NADH, you're going to be driving toward ketosis, okay, toward making ketone bodies. So that's a good thing to know in the process, because here we would need So so you would need NAD, right? So you would need NAD to go through the Krebs cycle, and then you end up making NADH. But if you have all this NADH around, this is going to shunt the pathway toward ketosis, making ketone bodies. Okay, so that's an important factor that comes into play here. And you can see here, so what's interesting, even through the process of starvation, you see you get to a point at about the 24-hour mark, you've like completely lost, you can't, you've depleted your glycogen stores in about 24 hours. So from the 24-hour mark of starvation onward, you have to be making new glucose, right? But you can see through this process, you're pretty much stable through the process of making glucose. And a lot of that has to come into play with the process of gluconeogenesis. You're just making new glucose through this process. But you can see over the time frame, look how many ketones have developed over that time. So beta hydroxybutyrate, again, is going to be the main marker you're going to look for. All right. Right. So, and this is kind of what we talked about. This is classic fruity breath odor, fruity odor on the breath. And you can see, look at their glucose, 400. So they have all this glucose around, like, why are you going through ketosis? Well, that's because they don't have insulin, okay? So you get this anion gap metabolic acidosis, or HAGMA is what they call it, high anion gap metabolic acidosis. And you also see this increased hormone sensitive lipase. And that is because they don't have insulin around, okay? And right. Again, you see hormone sensitive lipase is very active. Why is that? Because you are breaking down all those fatty acids. Okay, remember it's on glucagon's team, it's phosphorylated, it's breaking down fatty acids constantly because you don't have insulin. You can't use the glucose pathway. You're stuck in this ketosis phase. So hormone sensitive lipase is going to be elevated in ketoacidosis. All right, we can take a break. All right, we're making progress. I can't believe this thing's 600 slides. Get out of here. All right. I'm sorry. This was much easier when we were only doing like 1 or 2 terms. But having to or deciding to do this for all the terms has become very cumbersome. And listen, guys, we know this is painful, like all these pathways, and like I said, none of your patients are going to come in asking you about beta oxidation or anything like that. You know, we want to get to the clinical stuff. So we'll just trudge through and try to get through this. Anyway, all right. So cholesterol, right? Remember the main thing you learned back at FDM, it helps to stabilize that membrane. It actually stiffens the membrane. It's this big, huge, I think it's like a 26 carbon molecule. But among other things, right? So it's also a precursor for vitamin D. Remember, it's the precursor to those adrenal hormones as well. When you talk about that process, when y'all were talking about the CAH pathways, so it is that precursor to all those adrenal hormones. So yeah, but as for this is the same thing as ketosis, right? So, or for the ketone pathway, when they ask you what is the primary enzyme related in ketosis versus ketone formation, ketogenesis versus ketosis, you should have it, one enzyme involved in your mind, right? For each of those, same thing here. So the process of cholesterol synthesis versus cholesterol breakdown, okay? Yeah, and we'll get there. Okay, so HMG-CoA reductase, super important. That's where statins work, right? So when you talk about the number one or first line agent for stopping cholesterol synthesis, those statins, it blocks HMG-CoA reductase. They love to test on it because of that, because it's such an important drug. Okay, so know that. And I don't think they'll ask you about this Smith-Lemli-Opitz thing, but if they do, just know that it's in the cholesterol pathway, this mutation. I don't know, not really high yield. All right. And don't ask me why, no idea why this happens. They get a few digits of the toe because their cholesterol is messed up. I don't know. Right, now, what if we were to break down cholesterol? We convert it to bile acids, right? Stored in the gallbladder, stuff like that. That's why a lot of times when people have elevated cholesterol, they tend to get gallstones, okay? Because that cholesterol coagulates and forms stones in the gallbladder. So that's another good reason to use statins to help prevent cholesterol synthesis, okay? Why in the world did I put this on here? No idea. Oh, just the bile acid pathway. Oh, like what it's used for. Okay. Yeah, right. Okay. And here you go. So these are those cholesterol gallstones, cholelithiasis is the medical term for it. So again, you could see you get this referred pain. You remember you can get it also on the right side, what did we say, T7 through T9. You can get it if you have some sort of gallstone injury. When do you usually feel it? After a fatty meal, right? Because that's when that gallbladder is going to contract. Foul smelling diarrhea, why is that? Because if you don't have cholesterol, if you don't have bile acids or bile salts, you can't absorb fat. So if fat is in the diarrhea or in the stool, it tends to be foul swelling. They say that the stool floats, that means there's fat in it as well. You see that a lot with cystic fibrosis patients. So just in case that comes up on a question or on trivia night or something, you'll know it. All right. Now, we mentioned here, remember, depending on where the stone is lodged, will tell you what the lab values are. So they'll give you the lab values and they'll say, so let's say let's just go through it. Let's say this stone right here. What would you expect to be elevated? Okay, what are we blocking? Just the gallbladder, right? So we'll get an elevated GGT and ALP. Just to take note, you'll if it's a gallbladder problem, you'll never get you always have to have elevated GGT. How do I say this? Okay, put it this way. Sometimes in bone diseases, you can get an independently elevated ALP, okay? But if it is a gallbladder problem, you will get both an elevated ALP and GGT. So the GGT is really what's important, okay? Now, what if it's lodged right here? So again, you're blocking the gallbladder, so you're gonna get that GGT and ALP, but you're also blocking this path that's coming from the liver as well, right? So if that's blocked, you're gonna get an elevated AST and ALT, okay? What if you block it down here? Well, now not only are you blocking the gallbladder and the liver, but you're blocking the pancreas. So you're gonna get an elevated lipase and amylase as well, okay? So that's fair game. They can ask you that and just ask you where the stone is lodged. Cool. All right, what are we doing? Activated by insulin, hormone synthesis. Does that make sense? Oh, we have a hand raised. You could speak up, what's up?
Oh, I was saying sometimes they don't give you the enzyme levels, they just give you the symptoms that they present with. So
Yeah.
can we just say like, if,
Go for it, yeah.
So I was asking like symptoms, like solely gallbladder symptoms or solely hepatic symptoms or so.
Sure, yeah, and think about it, like if the hepatic ducts are blocked, like jaundice is a big thing that you're gonna get. If it's just the gallbladder, you're thinking of problems with the bile, so you get those fatty stools, and you get maybe even weight loss if it's long enough, and you'll get that that pain after fatty meals. So depending on that, but if they ever involve jaundice and stuff like that, that's a direct correlation with liver problems because of that bilirubin. Yeah, but thank you for mentioning that. All right, so if does that make sense that you're gonna make cholesterol in the presence of insulin? What are we? We're dephosphorylated, right? Insulin dephosphorylation. Of course, we're trying to store stuff. We're getting rid of all this excess glucose. Cholesterol is a good place to put it, right? Just like fatty acids, cholesterol. So again, this process makes sense. You would want to make it. Whereas if you're in a fasting state, you probably want to break down cholesterol, just like you're breaking down the fatty acids and stuff like that. Okay, so that should make sense. HMG-CoA reductase, right? All right, now same thing is for the opposite, right? If you're trying to mobilize and like bring up your blood glucose, you're not going to be making cholesterol. Like there's no point. That's not your goal at the time. You just want to bring up your glucose, right? You're not storing stuff. Here you go. Of course, statins, reversible competitive inhibitor of HMG-CoA reductase. Okay, we'll keep that in mind. All right, quickly, we'll go through this. Private Tim Hall, that is what everybody uses to remember the essential amino acids. The ones that have acid in it are acidic, the basic ones. Arginine and lysine, typically histidine kind of falls in that category, but it has like 2 pKa's on the chart, so it's don't worry about it, it's not important. Okay. Let's see. Right, so of course, the process of getting rid of nitrogen is very toxic in the body, specifically in the brain, but you're gonna, of course, make urea in the liver. Urea is basically a component. It has 2 nitrogen molecules hooked up to it, so it's a process of trying to put nitrogen in a form that you can get rid of. So again, you'll see patients, the urea has to be eliminated or you pee it out from the kidney. So in patients that have renal failure, you'll see elevated urea. That's a problem because it builds up in the brain. You get a lot of problems. You can get swelling or cerebral edema just because of all that urea in the brain. Okay, so just keep in mind that it's made in the liver and it's eliminated by the kidneys. Right. And then there's storage forms of it as well. I think we get into that in a little bit. Cysteine stones, so it makes sense if you have elevated cysteine in your blood or the cystinuria, you can form cysteine stones. Typically you see these in children until they're able to be diagnosed, and then you can get this, try to eliminate it from their diet. Okay, so these cysteine stones, I don't know that they'll really get into it too much detail. This is more of like a term 4 issue. So you see these cysteines are hexagonal. but I don't think this is really important for you guys right now. Heart-and-up disease, just remember heart-and-up deals with tryptophan. So you can end up getting those signs of pellagra or pellagrous at niacin deficiency. What are the signs of pellagra? There's like a triad, right? What is it? Glossitis. Anybody? Glossitis.
It's like the 4 Ds: dermatitis, dementia, diarrhea, and then death.
Okay, that's awesome. There we go. Yeah, I don't know what glossitis is. That's something else. My bad. All right, thank you. Yeah, the Ds. Oh, here they are. Perfect. Diarrhea, dermatitis, dementia. Okay, so that can happen with heart-and-up disease. What is glossitis? Is that a B2? I think it's B2 deficiency, then not B3.
Yes,
Anyway,
riboflavin.
Okay, thank you. Yeah, so those are the 3. I guess you can count death too, but diarrhea, dermatitis, dementia, pellagra, correlate that with tryptophan, correlate that with heart-and-up. All right, this is, I had to sit down with this for a while, and like I actually put the nitrogens in here because the whole point of this, I think this is Dr. Sobring's lecture, so I don't know who taught it for you guys. But the process is, how do we get rid of these nitrogens? Like, you can't just have nitrogen floating around in the blood. You have to store it as urea. You have to tag it on one of these amino acids. You have to put it somewhere so it's not doing trouble, okay? So if you follow the nitrogens, you can see how it works. So alanine has a nitrogen on it, and it uses this process of converting to glutamate to make pyruvate. But the point I want you to see is that the whole point here is that we are transferring these nitrogens around until we can get it to urea, until we can get rid of it. Okay, so this is that process, not super high yield, but you can see how this works. Same thing here. So, and you can see how this fits into the Krebs cycle, right? You see how pyruvate, oxaloacetate. But the point here is that, look, we have 2 nitrogens on the asparagine. We go through this process, aspartate, it's 1 nitrogen because we put it as ammonia. And you could follow it around. So alpha ketoglutarate does not have a nitrogen, but when you combine it with aspartate, or I'm sorry, when you take aspartate and you put a nitrogen on alpha ketoglutarate, you get glutamate. Okay, so it kind of all just like fills in the gaps here, and then you get oxaloacetate. So then you can go through the Krebs cycle. Okay, so that's just the point I wanted to get across, is that you can actually get rid of the nitrogens here, which is kind of the whole point of doing this process. Okay, again, you can see glutamine, which is actually where it's stored in the brain. So patients with renal failure, patients with elevated urea, they end up getting a lot of glutamine because you're trying to store this nitrogen somehow. So the glutamine actually is elevated in the brain with all this nitrogen, and you get cerebral edema because of it. It's like an osmotic pressure that develops because of this glutamine. Okay, so again, you can just see this process. I don't think this is like something necessarily you need to whiteboard out. I just wanted to get across the importance of being able to deal with all this nitrogen. Okay, so this is kind of what I said. You can't have it. You need to be in this non-toxic form. So if anything, just know that the glutamine is incorporated, same with the alanine. Okay. Right, so yeah.
They wouldn't have you pick between either of those, right?
No, probably not. Maybe just the fact that alanine is specific to the muscle.
Okay.
But I don't think so. I don't think this is very high yield in general. So, but no, I don't think they would. Okay. Right. And you can see here, this is the urea. You can see it takes 2 nitrogens, hooks it up, and then you can get rid of it in your urine. Urea cycle, it's a pain, I know, but there are some diseases that are correlated with this. Right, I think there's a better, oh, here they are. Okay, where's the, oh, sorry. So this is the reactions of it. I think if anything, just know these. Some of the differences here, if I remember, arginemia does not have elevated ammonia. The rest of them do. This one's important, this ortic acid that develops in type 2. And then I believe type 1 is the most common. So they like to test on the common things. What else? All of these kind of fall in line except the citrullinemia, because it uses a different enzyme, right? So like arginemia is arginase deficiency, or this one is this, but citrullinemia has a different enzyme involved, okay? But you do get elevated citrulline. So these questions should be fairly straightforward. Like you should be able to answer it if you know this chart. I wouldn't necessarily bother writing out the pathways and stuff. Very important for these patients, the treatment that you can give is phenylbutyrate. But actually, we did have this question. We had a question and it was phenylacetate. So phenylbutyrate, it's like some sort of breakdown product or, oh, it forms phenylacetate. Yeah. So they actually use phenylacetate on the test as a treatment, not phenylbutyrate. So they're synonymous for all intents and purposes. You could also use benzoic acid. Okay, so this will help mop up all that extra urea. Right, and this is what I was talking about, the cerebral edema that forms from all that glutamine, trying to hold on or keep that nitrogen out of circulation. All right, very busy, but that's basically what I said. You get all this glutamate to glutamine and yeah, glutamine levels. Right. So this is PKU, the process. So definitely, of course, know the enzyme that's involved. You need to take this out of the child's diet. Otherwise, they can get neurologic deficits and stuff like that. Yeah, they do test it for this at birth. It's that problematic to the intellectual development of the child. So they actually test at birth to see if these, at least in the US, they test to see if you have PKU. Yeah, low phenylalanine diet makes sense. You know the process or the pathway: phenylalanine to tyrosine, dopamine, norepinephrine, epinephrine, right? So you need to get rid of the phenylalanine, but of course that means the next step in the process, because you do need those amino acids, that means tyrosine becomes essential, right? So you're bypassing this tyrosine, this phenylalanine hydroxylase deficiency. No phenylalanine, you need to go ahead and give the child tyrosine, okay? And there's that other one, I don't know that it's super important. It's not the classic PKU, it's the other one. I don't know what it's called, like PKU2, but it has to do with these not cofactors, but these coenzymes that are used. Yeah, I might have a slide on it, but this is the main ones that they talk about, the classic PKU. Right, maybe know that, I guess. I don't even think, they use it for some PKU patients. Just keep it out of their diet. And of course, if the mom, wait, how does this work? Yeah, so, okay, right. So even though the child doesn't necessarily have PKU, if the mom has PKU and has elevated phenylalanine levels, it can be toxic to the child. It can pass through, and the child pass through the placenta, and the child can get intellectual disability. So the mom has to be aware of that as well. Alkaptonuria, right? So homogenistic acid oxidase is the enzyme involved there. Still the same pathway, this phenylalanine pathway. And you can see this, the urine turns dark on standing, it's oxidized. So that's a buzzword for it. You know what we're talking about there. You also get this darkening of the connective tissue as well. Okay, tyrosinemia, not super important, but yeah, the cabbage-like smell, that's whatever cabbage smells like, I don't even know. But yeah, that's what they say. Tyrosinemia. And then maple syrup urine disease, of course, their urine smells sweet, which is funny, actually. I asked people about this. I'm like, does it really? And they're like, not really. Like, it doesn't, I don't know, like, I don't know, I made this stuff up. But for the test, just know that it smells sweet, apparently. Or like fruity breath odor. Like acetone doesn't smell like fruity breath. Not that I'm going on a tangent, but it doesn't smell like it. And people say that people in DK, their breath doesn't actually smell fruity. So I guess for the test, just know it, but I guess it's a real life problem. All right, so neurologic problems, they can go into ketosis, right? And have this maple syrup urine smell, okay? And this is that other one. So we talked about alpha-ketoglutarate and pyruvate dehydrogenase deficiency, but don't forget this branch chain alpha-keto acid also uses these coenzymes, TLC for Nancy. Okay, vitamin B12 problem. How do you know the difference between vitamin B12 and folic acid deficiency? Remember, they both cause megaloblastic anemia, those big red blood cells. The difference is when you have vitamin B12 deficiency, you're going to get neurologic problems, okay? So that's the only real way that you could tell clinically from it. And but when you look at their urine, they also have a buildup of this methylmalonyl-CoA, and that's because you need vitamin B12 as a cofactor. So if you don't have it, this gets built up in the urine. Okay, so if you're trying to differentiate folic acid from a B12 problem, the neurologic stuff and this in the urine is diagnostic. Okay. And you can see this is not very common either. I don't know that they really will test you on that. Right, homocystinuria, you can see this is this cystathione beta synthase problem here. And these children look very characteristic, look to them. Dislocation of the lens, skeletal abnormalities, delays in mental development, kind of sounds a lot like Marfan's, right? Even in presentation. All right, for the record, a lot of people had trouble with this, and I don't know if there's an easy way to go about it, but I can tell you that it wasn't super high yield, this physiology going through like the different motilities and stuff like that. What they really tested on were those like CCK and all those hormones that are involved. So that's what I'm gonna key in on. Some of the differences here, like the myenteric plexus is involved with motility, where the submucosal or Meissner's plexus is involved in secretions. So primarily a parasympathetic response, but you can keep those in mind. We talked about this. Remember, failed migration of the neural crest cells. Okay, and then of course there are different ways of regulating it. The primary one we talk about is the endocrine secretion. So all this is fair game. Know the cells that go along with it, know what they do. Somatostatin's a weird one because it's like, wow, okay, so it blocks insulin and glucagon. And I was like, well, what's the point? Like, what are you doing? Like, you need one or the other. But what actually happens is that in the stomach, sometimes, the stomach needs time to digest the food and the acid. So the idea of somatostatin is that if you block glucagon and insulin, and you actually block the gastric motility, it actually allows the stomach to kind of sit there with the food and kind of like let it digest. So that's the idea there. So that helps you kind of keep it straight. Of course, gastrin, the G cells, remember, and then that has a lot to do with those parietal cells that come along, but we'll get there. Zollinger-Ellison syndrome, that's a good buzz or a good disease to know, like gastrinoma, so they'll present with really bad ulcerations, right? Because of all this gastrin and acid that's developed. Let's see. I remember the difference between ghrelin and leptin. Like ghrelin sounds like grr, like your stomach's growling. So it makes you have an increased appetite, whereas leptin is just satiety hormone, which means it kind of tells you when you're full, okay? Right, and then we know intrinsic factors very important in the parietal cells. We have those deficits like autoimmune problems, destruction here. So if you have that in the parietal cells, not only do you not get acid that's produced, but you also don't have intrinsic factor. So you can't bind the vitamin B12 to carry it to the ileum to be absorbed. So remember, the whole point of B12 is once you ingest it, it binds to this intrinsic factor in the stomach that comes from the parietal cells, and then it goes through the gut to the ileum, and then that's where it's absorbed. So if you don't have parietal cells, you won't have intrinsic factor, and you also won't make hydrochloric acid. It won't be responsive to the gastrin that comes through. All right. This kind of outlines where everything is. I don't know if the day before the exam, this is something you want to worry about, but yeah. Right. So of course, acid's going to help to regulate, gastrin's going to help to regulate your acid secretion. So that's super important. CCK, remember, after those fatty meals, CCK is going to be directly involved with gallbladder contraction. So they could ask you a question. They could say, patient has a fatty meal and they have right upper quadrant pain, and they may not tell you that the patient has cholelithiasis or any sort of problem, but they may ask you what is the hormone that's involved in this pain. So CCK helps to contract the gallbladder and relax that sphincter to allow the bile to come through. So if it's not, if the patient has pain and they have stones there, it's CCK is the problem. All right, and then secretin as well. Yeah. Okay, yeah, right. So it reduces the acidity of the small intestine, bicarb, right? So it counteracts gastrin. And then these as well, these aren't super important. I don't think we had any questions on this. Just have a definition for them. And then some of the paracrines, so that means they're actually found in the stomach. So somatostatin, like I said, kind of helps to let the food kind of sit in the stomach for a little while. And then histamine is also there in the stomach, increases gastric acid. Then some of these other ones that are a little lower yield, right? For some reason, this is something I just skipped over, and I think we had a question on the exam. I think it was this right here. It said like, what receptor did gastrin work on? And it was this one, CCKB. So I would know these, of course, vagus nerve being parasympathetic at this point would work on the M3 receptors. Histamine you would expect to work on the H2, but they asked about gastrin in this, so make sure you know that. All right, we talked about leptin and ghrelin. All right, don't make this more confusing than it needs to be. Manometry is just a pressure wave, all right? You're measuring pressure. So you should expect more pressure in the upper and lower esophageal sphincters, okay? So that's what we're looking at here. If you were to look at this, see, this is the point at which you swallow. Remember, it's like a pressure wave that goes down. So you could see at the point where the patient swallows up top, you hear the pressure wave, and then it kind of, over time, it goes down, okay? And then after you get this reflexive, after you get this swallowing, you get this reflexive relaxation after, okay? So they may give you a question like this and just ask you to interpret it. They may just give you the wave down here and like not show it contracting at the lower esophageal sphincter. What could that lead to? Stuff like GERD, right? You need this sphincter to be contracted, otherwise that acid's going to reflux up into the esophagus. All right. Yeah, so there's different types of contraction. Segmental contraction kind of just helps to get the food all together, where then you have, when at that point, then you get peristaltic contraction, where everything moves down. Right, and then of course, smooth muscle goes through this tonic contraction, but you also get basic contraction, and that allows for the food bolus to move unidirectionally. Right, so of course, acetylcholine is a parasympathetic response. So increased contraction and motility, and it says here opposite. So BIP and nitric oxide will decrease that. Okay, so there's a balance here. Again, these are hard to write questions for. So a lot of this stuff just wasn't really pertinent. This migrating motor complex, it is like when you're not eating in the fasting state, it just kind of helps. It's just like a low grade movement of the gut, and it just helps to prevent this stasis of food in your gut so you don't get any sort of infection. But most of the time, at least after you eat, you get this segmentation and peristalsis process. Okay, and then that's a good thing to know. So this migrating motor complex, motilin is responsible for it. So again, that just prevents stasis of food and bacteria and stuff like that. Not super important. These slow wave contractions from these interstitial cells of chyle. Yeah. So just the basic electrical rhythm, that pulsation wave that keeps everything unidirectional. Right, and this is the idea. So these slow waves are always going, but it's not going to reach threshold. So once it reaches threshold, that's when you actually get the peristalsis, the slow wave movement. But the idea is they're kind of always on the verge. And then once you eat or once you get some sort of stimulus, then you get a full action potential. Again, not super important. It's really hard to test on stuff like that. Right, and then again, so this is this fasting state, you still get these migrating motor complexes. Motilin's responsible. Right. Okay, that's pretty straightforward. Large intestine, same thing, right? Mass movements, that's what you would expect. And then this shuttling or segmentation makes sense with these hostra that are formed. Whoops. I don't think they asked us anything like this, like time frames. But as you move down, the process gets slower, right? So more food will be in the rectum, or fecal matter in the rectum or time frame. It'll stay. The further you move down the digestive tract, the longer it'll tend to stay. the only thing they asked us about these reflexes, was this gastrocolic reflex. So let me just explain. The idea is that if you ingest food, this is the way for your stomach to tell your colon that there's more food coming in. So if there's more food coming in, some food's going to have to come out. You're kind of telling downstream, like, hey, we're bringing more food in. So that's the one they asked us about. They all kind of work similarly in that regard, like the upper GI tract telling the lower GI tract that, like, be aware of food's coming. But that's the one that they asked us about. All right, pretty straightforward. All right, this is the pain. I don't even remember this, how this works. Which one's which? Okay, so the more time, okay, so the more time it spends in the duct, the more hypotonic it gets. Okay, yeah. So if it comes out of the duct, immediately it's isotonic, which I guess that kind of makes sense, right? Because, like, you don't have time to shuttle everything out. Okay, so if it spends more time in the secretory duct, you're able to reabsorb this sodium chloride, and you're able to put more potassium and bicarb in it, and it becomes more hypotonic. I don't think they could really ask you anything beyond that. I know the pancreas is a little bit different, and this is kind of saying the same thing. So the longer, the flow rate is slower over here, and it's faster over here. Okay, so that'll determine, see, I put this over here. Hypotonic, slow, we're on this side. Fast, you don't have time to move stuff around or take stuff out of secretion, so it's fast. But it's tricky because the pancreas is like opposite, but we'll get there. Alkaline tide, right? That's very important, because if you have all this acid, if you're stimulating this acid secretion in the stomach, you need to have bicarb to offset it. So you get this alkaline tide that's formed, and that's the whole goal, is to counteract that acid. And then, of course, cephalic phase, when you think of food or smell food, right, you get that, and then you get to gastric and intestinal phase as well. And this is just that balance, right? So again, any sort of acid that develops, you need bicarb to offset it. Mucus helps as well, stuff like that. Prostaglandins help with your barrier as well. That's why people that take aspirin too much, that'll help. That actually blocks prostaglandin synthesis, or NSAIDs in general is probably a better term. And if you do that, then you're not going to have a barrier, so you tend to get ulcerations. Okay, and then some of the drugs, I don't know that they'll really ask you about this, but they're here. I would just know which one's working, which pathway. Particularly this omeprazole is Prilosec. So yeah, it's one of these potassium ATPase inhibitors, or this proton pump inhibitor. And then pancreatic secretions, of course, CCK, secretin are going to come out of those cells from the duodenum. But primarily through the pancreatic secretions. I mean, not only do you get those prohormones like trypsinogen and stuff like that that are formed, but a lot of this is going to have to do with bicarb. There should be a better picture. So bicarb, chloride, this is it, right? So this is a little different from the other one. If we look at our slow flow rate or low flow, it's going to be mainly sodium chloride, and high flow, you're getting this bicarb. So just remember, the faster the flow, the less time it has to be modified. So if there's less time, there's more bicarb. If there's more time, keep the bicarb, and you get sodium chloride. Okay, y'all know about cystic fibrosis. We don't have to go through that. This is very busy, but it's still talking about the same thing. So again, if you're worried about this physiology stuff, I would look at first aid. I would just know those tables. Just know basically these hormones and how they work and where they work, and you should be good. That's what we were tested on. All right. Most of the absorption obviously goes through the small intestine, and there's also some in the large intestine if need be, mostly water. All right. Osmotic, water, secretory, of course, has toxins and stuff like that. Some of that. And of course, with diarrhea, primary problem is you lose a lot of bicarb. So you end up with some sort of metabolic acidosis with that. Whereas when you vomit, you throw up a lot of acid, so you get metabolic alkalosis. So they're opposites. Oh, sorry. And again, if you have diarrhea, you also lose a lot of potassium as well. So you can end up with hypokalemia. So dehydration, hypokalemia, and metabolic acidosis because of all of the bicarb that you lose. Right. And it says that here. Great. All right. So this is Dr. Trotz's stuff. You kind of just read through this. She puts everything in. Everything you need to know on there. So of course, in the mouth, you have these salivary amylases, and you swallow the lingual lipase, get down for lipid digestion in the stomach. Kind of talked about those. So CCK, not only does it contract the gallbladder, but it helps enteropeptidase in the duodenum, and with the secretion of these pancreatic enzymes. Zymogens are just those pro-enzymes like trypsinogen. Once it's a zymogen, and then it's cleaved to trypsin. Okay, so that makes it active. Once trypsin's formed, it's like this, all hell breaks loose once trypsin's formed. Trypsin's that first one. Once you take trypsinogen and you cleave it to trypsin, then trypsin activates everything. This is the problem that happens in patients that have cystic fibrosis, because remember that bile duct is clogged with mucus. So when the pancreas tries to get rid of all of the zymogens, it can't get out. So this trypsinogen is cleaved to trypsin in the pancreas, which is not supposed to be, and then you get autodigestion of the pancreas. Again, it's because that duct is clogged and nothing can get through. Okay, kind of outlining big picture stuff, and here you go. You can see everything's cleaved. Remember, CCK will activate enteropeptidase, then trypsinogen's formed, and then it's activated to trypsin. More words. Kind of talked about bile, mostly to reabsorb fats and fat-soluble vitamins A, D, E, and K. Right. Yeah. Right, fatty stools, again, typically a bile problem. Cystic fibrosis is when you see that, okay? Again, and that's because you can't get those pancreatic enzymes or that pancreatic juice through the duct because it's clogged with mucus because of those CFTR problems. All right, we talked about stones. Yep, remember, depending on where the stones lies, it'll tell you the lab values or the clinical symptoms. Let's see, there was, so, where's that? I think, oh, here it is. Yeah, let me just, okay. So the bile salts emulsify, okay, right. So they're conjugated, the bile salts will conjugate them. And then once they get to the bacteria, they're deconjugated so they could be recycled. Yeah, sorry, I have to relearn this. This is the slide I wanted to show you. So again, it's bile acids, and then they're converted to bile salts. And then once they're converted to bile salts, they're active and they could work with absorbing fat and vitamins and stuff. And then once they get to the intestine, the bacteria can convert it back to bile acids, and then they're recycled again. Okay. I think that's all. Primary and secondary bile acids are conjugated with glycine and taurine. Okay. Yeah, you can see they put the on here. And are released as bile salts, right? And that's the active form of them. So again, after they're made from bile salts, the bacteria can convert them back to bile acids or deconjugate them, and they're converted back and brought back to the liver to be used again. Okay, Lindsey, it's your stuff.
Okay, let's do this. Okay, here we go. Oh, hey, can you make me co-host again? My internet caused me to drop out for a minute and I can't share my screen.
Sure.
Thank you. Okay. Gonna run through this quickly. So the papillae, lingual papillae, I think there was like maybe one, maybe two questions on this. This is kind of a lot of information. Just be able to tell the difference between the four of these. So filiform, fungiform, folate, circumvallate. Big one, circumvallate. This one's quite easy to recognize. The big thing, receive the ducts of the serous von Ebner's glands. The foliate has taste buds, so does the fungiform. So just make sure you can identify each of these. Remember, if you have multiple, you will probably see each of these on there and you'll have to pick one. Okay. So esophagus, so going down the system, stratified squamous, non-keratinized. You have a lamina propria with cardiac glands that produces mucus, and then the muscularis mucosa. This is important because the presence of cardiac glands is going to be important, and then muscularis mucosa, of course, because esophagus, you're going to have peristaltic activity to propulse the food bolus down. This junction, this is bigger for, no, it is histo. So at this junction here, you can see that there is a clear delineation. And so this also means that you can have some pathology here because you can get some metaplasia. You know, they love metaplasia. But so stratified squamous to the simple columnar when you're getting into the stomach. Cardiac glands, you're now getting into the cardiac glands of the stomach. The muscle, so just be able to recognize that. I'm trying to find the high yield. Okay, Barrett's esophagus and GERD, this is very high yield. Metaplasia, again, anything that they talk about metaplasia is going to be high yield. And so, you know, it's going to be your typical vignette where they go in, they have some symptoms, to take a biopsy, they show cells that are changing, and then you remove the stimulus, and then a few months later it's normal and it was metaplasia, that kind of thing. Stomach, this is going to be big, the different areas of the stomach and what are in those areas of the stomach. So cardiac regions up at the top here contains cardiac glands. Fundus, you have the gastric glands, and then the pyloric region contains the pyloric glands. So just this is bigger in physiology, but just understand the functions of it, mixing, digestion. You get very minimal absorption, and the reason you have this is because of the pH. Remember, back in FTM, they talk about acids and bases being absorbed into the system and what part. So cardiac region, you have short pits and short cardiac glands, whereas pyloric, long pits and short pyloric glands. Do know how to tell the difference between the two of these just looking at the slide. So, you know, what's highlighted in blue is how you're going to be able to do that. Cells, you mucus secreting cells, and you have the enteroendocrine versus pyloric, secretory, and then enteroendocrine. It is very, very, very important that you understand the cells that are in each of these regions and at what level they are in the glands, because the cells lining the glands towards the lumen, they're going to be different than towards the submucosa. And so understand those, understand the type of cells and those secretions. That's a very high yield point that they will ask you about. And they'll ask you about it in more of a third, fourth order kind of way. It's not going to be first and second order. So fundic region, again, know how to tell the difference, short pits, long glands, and then cell types found, mucous neck cells, parietal, chief, enteroendocrine stem cells. So I want you guys to star this slide, highlight it, and the subsequent ones that talk about these, because these are going to be very important and you do need to be able to recognize them. So we'll go through them. So you do have hydrochloric acid produced by the parietal cells. It's very important. So converts inactive pepsinogen into pepsinogen. That is important. So pepsin is produced by the chief cells. Then the mucus, you're going to have mucus that is, I forgot what secretes that. And then intrinsic factor is by parietal cells. This is very, very, very big because this is going to bind B12 and this allows for absorption. So if you have destruction of parietal cells, you are not going to have intrinsic factor. Therefore, you're going to get a vitamin B12 deficiency. Vitamin B12 is absorbed in the distal part of the ileum. So kind of put all of this information together to understand how they could ask you a third or fourth order question just about this one type of cell. So the mucus cells, surface cells, remember I said, please know where they are located in the gland because they're going to be different from top to bottom. So the mucus cells are the top, they secrete a viscous mucus. This makes sense that they would be towards the lumen because the lumen is the part of the area that's going to be subjected to everything all the time. So you want a protective layer. So you're gonna have the cells toward the top because they're secreting mucus to protect that area. And then mucous neck cells. So surface is thick mucus, and then mucous neck cells, it's soluble mucus for transport. Chief cells, very high yield. Know what they look. Know what they look like, please. So these are the more basophilic cells versus the parietal cells, which are going to be more eosinophilic, and they're kind of mixed down here. So make sure that you understand, like if I zoom in here, oh, wow, okay. These basophilic cells are going to be the chief cells versus these more eosinophilic ones that are kind of, I call it a fried egg appearance. I don't know if they call it a fried egg appearance, but those are going to be your parietal cells. So if you were given this slide, please make sure that you can tell the difference just by looking at it, which are the chief and which are the parietal. Again, it's going to be more third or fourth order. So they're going to give you a vignette. They're going to point to either a parietal cell or a chief cell, and they're going to ask you something about that. So it's going to be more along the lines of the secretion and what happens if it's damaged. So for example, like I just said, parietal cells, if you damage those, you don't get intrinsic factor B12 or hydrochloric acid, and you don't get the B12 absorption later on. So just those are two very, very, very high yield points. And then enteroendocrine, this is another high yield one. So secretes hormones. So gastrin secreting cells concentrated in the two general types. Oh, so you have closed and open, and then the gastrin secreting cells. Okay, so just still moving down the system, you have the gastroduodenal junction. So disappearance of the innermost oblique layer. And now you have, again, the two typical layers of muscle going into the small intestine. Peptic ulcers, so incredibly high yield, so incredibly high yield. Anterior versus a posterior perforation, what it looks like when you get a perforation. Yes, you need to know both of these images and that they are associated with peptic ulcers. Its association with H. pylori. the secreting urease and proteases, that's more of a conceptual thing, but you know, understanding what it does, but you definitely at this point need to know its association with H. pylori. If you see one of these pictures, you know what could have caused it. Others, tobacco, NSAIDs, and then gastronoma. Those are, yes, be aware of those. H. pylori, they love to bring up those because it's such a common cause of peptic ulcers. So you no longer have any protection, you're getting inflammation, the tissue can become necrotic, you're leaving a sore ulcers, and then, yeah, you get fibrosis and scar tissue over it as well. So note those two images, know its association with H. pylori. Okay, now moving down, small intestine. This is just an intro slide, so I'm not going to spend too much time on it. So this histo picture, please, please, please note it, know what it's called. So you have mucosa with so simple columnar, okay, you have galt, which is important, but the big thing, you have villi, which are the invaginations, and then you have the crypts of Lieberkühn. So these are going to be the invaginations of the epithelium into the lamina propria, and then the submucosa, you're going to see the plicae circularis. So on this histo picture, you see, so do I have, where's my, I don't know, annotate, here we go. So this right here is going to be your villi, but this entire thing, the PC, is going to be your plicae circularis. And so see a submucosa right here. You see this is an invagination of the submucosa, whereas the villi, it's just the epithelium and the lamina propria. So understand the difference between those two. Know that this individual one right here, the invagination of the epithelium lamina propria, that's just the villi, and then the plicae circularis is this larger invagination of these submucosa. And of course, every time you do this, every time you do a little fold, you're increasing the surface area because you increase it for absorption. Okay, so we talked about the plicae circularis, so most numerous in the distal duodenum and jejunum. So this forms a characteristic appearance of this area. Villi, these are the epithelium and lamina propria, so these are small one, and then of course microvilli. So we're getting, so from 1, 2, 3, we're getting larger to smaller essentially, and we just keep increasing the surface area. And then the microvilli, this is what is referred to as the brush border. Remember, this is where you have all of those disaccharidases that are going to break down the sugars. And so if you have something wrong with the microvilli, you're going to get a lack of absorption or intolerances. Okay. Intestinal glands. So these crypts of Lieberkühn. Move that out of the way. So an invagination of the lamina propria, so that's at the level of the villi. Simple columnar, continuous epithelium. I don't remember why this was significant. Paneth cells, I remember that being a thing. These Paneth cells, that was high yield. Circle that, highlight that. Okay, more cells. So we have, oh, we're going to talk about Paneth cells here. Cool. Cells, enterocytes. You have the goblet cells. You have your Paneth cells, enteroendocrine. The Paneth cells, the big thing, yes, you need to know how to identify these. So you'll see that you can see that there are a lot of what looks like granules in here. So you have lysozyme and alpha defensins. That is important. Know that those two are within the Paneth cells and how to find them. And then the goblet cells as well. So mucinogen granules, accumulating, and then increase in number from duodenum to colon. Know that as well, their location. If it tells you a certain location, that's definitely going to be high yield. And then enteroendocrine found at the base of the crypts. That's a big thing there. Most active regulators released in small. So enteroendocrine cells are going to be responsible for that, the CCK, secretin, GIP, and motilin. Okay, moving along duodenum. Please, please, please know the different specializations in each of these. If you see these Brunner's glands, it's very, there's a lot of these glands in the submucosa. They absolutely love to test this for cumulative questions. So on your NB exams, they're probably going to give you a couple GI histo questions, but they're not going to be, they're not going to try to throw you for a loop. Do you know the specialization in each area or don't you? So in the duodenum, you have these numerous Brunner's glands in the submucosa. So highlight that, star it. That's the huge distinguishing factor. And it's very high yield. Please don't underestimate that information. And then in the jejunum, you are seeing these plicae circularis, so long prominent villi. Know that. Ileum, you're seeing these Peyer's patches. Again, very high yield, highlight it. And this is also the ileum terminal portion. This is the absorption of B12. They'll probably relate that back to the whole intrinsic factor thing. So just understand that. You will have an increase in Panethin goblet cells. So if we look back at that, there is a significance there. And then the Peyer's patches, that is the immune aggregates, the lymphoid aggregates there. But the Peyer's patches is what's going to be the thing that clues you in on I'm in the ileum. Celiac and Crohn's, they will probably ask you one question, maybe two questions on here. Just know the difference. So celiac is gluten intolerance. Autoimmune mediated diagnosis, you have IgA antibodies for transglutaminase and endomysium. The deamidated I don't know how to say that, just highlight it, it's important. And then Crohn's disease is ulcers, so ulcer formation in the small intestine, and you get malabsorption. Cobblestone appearance, highlight this, the cobblestone appearance. This can be something that clues you in on Crohn's disease. And then patchy distribution, which is called skip lesions because it's not uniform. Then the long intestine, ascending, transverse, descending, and sigmoid. This is just the areas. Tinea coli. So this is just bands. These are longitudinal bands. So you see this right here labeled TC. This is just a longitudinal band that extends the entire length. Then the hostra. These are the visible sacculations. So you can see this when, if you're doing a barium enema and you do an x-ray for diagnostic purposes, you can see that because it'll look like these indentations. And then you can have elemental appendices, fatty projections on the outer surface. So the two biggest ones are the Tinea coli and the Hostra coli here. The mucosa in the large intestine is going to be smooth, so no villi. This makes sense because you don't have large amounts of absorption happening in the large intestine, but you do have numerous glands. So you're reabsorbing water and electrolytes and eliminating waste. But remember, we don't have any villi because this isn't the main site of absorption. You are getting absorption, but it's not the main site of absorption where you're trying to take everything into your body. The absorption is more like regulating water and electrolytes instead of give me all of the nutrients so that I can survive kind of thing. No panicels, that's important if there is an absence of or an abundance of a type of cell or a gland. Definitely make note of that. It's going to be important. But the muscularis externa, the muscles in the digestive system are very important because this makes up the different plexuses that control movement through the digestive system. So very, very, very important. If you have disruption of any of those, you have really bad issues. So toxic megacolon, that is going to be a, I believe it's the myenteric plexus that is not innervated. I think that's what it is. But you have a lot of issues here if this gets damaged or if it's not functioning for any reason. But just understand that. So muscularis externa, it's found in all areas, inner circular and outer longitudinal, and then you have the myenteric plexus. Appendix, this picture is very characteristically, like I look at this and I see appendix. So just please understand, or please look at this and see appendix. Thin finger-like, so tinea, you do have tinea coli extending at the base, so it can help you identify this. And then numerous lymphatic nodules extending into the submucosa. So this is its defining characteristic. So you see the lymphatic nodules that are extending. Okay, anal canal, there are different areas. I would spend a few minutes on this slide knowing the difference between the two areas. So the colorectal, you have simple columnar, and then you get the anal transition. You get, the simple columnar, and you're transitioning into stratified squamous, and then you get to the fully stratified squamous keratinized epithelium. That could be something that they ask you depending on which area you are in the anal canal. Okay, so this is what I was talking about. Congenital megacolon. I call it toxic megacolon, but congenital megacolon, Hirschsprung's disease, they love to test on this. This is a very high yield. So failure of migration of neural crest cells. And so you don't get development of the myenteric plexus. And this is responsible for the peristaltic movements that are going to move everything through the system. And so it's just not moving and it's not being released. And so you get a very, big obstruction, usually kind of in the sigmoid colon and the distal parts. And so you see this very characteristic ballooning right here in this cartoon image. Okay, salivary glands. I'll touch on this in FTM1. So parotid glands, simple, classified as pure, acinous, compound, alveolar. Okay. Yeah, so a very high yield point opens into the oral vestibule at the parotid papilla, so next to the upper second molar. Highlight that, start, you need to know it, I took this piece of information for granted on each of the glands and I regretted it on the exam. It's only 1 or 2 questions, but at the same time, it's like, oh, that 1 or 2 questions. So that's very high yield point. Same with submandibular, opens on either side of the base of the lingual frenulum. So this is going to be mixed gland, compound tubulo-alveolar gland. Sublingual, same thing, directly onto the floor of the oral cavity and into the submandibular duct. Mixed compound tubulo-alveolar gland. But make sure that you can put these side by side and tell them apart. And so if they didn't give you the gland and they just put it side by side, you would be able to, okay, this is this type of gland. And now where does it open up to? So wherever it opens up is very important. So remember, parotid is just pure. So parotid just pure serous. But I don't know about you, but when I think pure serous, for some reason, I want to picture more, let me annotate. I want to picture more this because it's like clear. I mean, that's duct, but I picture something more clear for some reason. I don't know, my brain just makes that association. But the pure serous, it's this more eosinophilic that you see right there. And so, okay, it's pure serous. I'm in the parotid gland opening up next to the upper second molar there. And then submandibular and sublingual are both mixed, but one of them is more one thing than the other. I don't remember which is which, but this is okay. So this is mostly going to be the mucus and then some serous, but yeah. Brady, is this back to you?
Looks like it. Let's just take a 5-minute break and then we'll finish up. All right, let's finish quickly. All right, let's see. So remember one carbon metabolism, like SAM, remember that SAM is that one that transfers methyl groups. SAM's your meth dealer. So that's how you transfer those 1 carbons. But in regards to some disease state, I did mention this earlier. This isn't the classic PKU, but this is that other one that has to do with those cofactors, the BH2 or BH4, the dihydrobopterin reductase, right? So that could come up. It's just worse. It's more CNS symptoms that you could see there. Likely they'll test you on the classic one, but just in case that comes up. And you could see this pathway. So not only will this affect this phenylalanine hydroxylase, but these downstream effects too, that it's a cofactor for as well, right? So let's see. Yeah, so it will affect all these pathways, which is why the symptoms become more severe. Y'all know both of these by now. Yeah, hypopigmentation, there's a good buzzword or a good clinical situation that'll help you isolate that. Right, we talked about SAM, methyl dealer. We talked about, did we talk? Yeah, we talked about homocysteine. Remember, they kind of look like those marfanoid habitus, or look like they have marfans. So this is that process, it's the thione beta synthase. And then homocysteine, you kind of have to get rid of it. So you convert it as soon as you make it, so that it's, because if you get elevated levels of it, it is toxic, right? So it is required to make some of these cofactors, but I'm sorry, it's like folate and B12 and B6, but again, you don't want homocysteine floating around. You want to be able to break it down, right? So you can either make cysteine out of it or back to methionine. Okay, so, right, we talked about this earlier, megaloblastic anemia. How do you tell the difference between folate and B12 deficiency when the cells look the same? You look at the symptoms. If you have B12 deficiency, you will get neurologic problems. Also, methylmalonyl-CoA will show up in the urine, right? Because that you need B12 to break that down. So that is characteristic of B12 deficiency. Yeah, and then FIGLU, yeah, that's the folate deficiency. So if you don't have folate, you can get elevated FIGLU. Just something to know. I mentioned neurologic features. So some of the problems that to develop B12 deficiency, one of the big ones, especially since you're doing DM right now, is this lack of intrinsic factor from the parietal cells. It's called pernicious anemia. That's what it's called. And you have autoimmune antibodies that go against these parietal cells, so it kind of kills them. So you don't get intrinsic factor, you can't bind B12, so you can't bring it to the ileum. So you can't absorb it, right? So these are some other factors that can cause it. Gastritis will do it, and any disease of the ileum, even a resection of the ileum. That's a good one, actually a good test question. They say somebody went through an ileal resection, you're not going to be able to absorb B12 because there's no ileum there. So you can get symptoms of B12 deficiency. Right, okay. Methylmalonate or methylmalonyl-CoA. That's what you'll see in the B12. All right, heme metabolism. This is from first aid. This is Dr. Mpadja's slide. So this is one of those ones you kind of have to know all of the whole pathway, because there are different pretty much every step here has a problem that can develop from it. So make sure you can get those down. Okay, we're gonna go through it. All right. So B6 is a cofactor here. Yeah. So allosynthetase, if you don't have that, you can't make this aminolevulic acid. Let's see what it says. So you can't start it. Okay, yeah, so isoniazid is one of those drugs that they use for tuberculosis. Yeah, and it'll cause B6 deficiency. So that could be a problem. Lead poisoning, lead will inhibit zinc, right? Is it zinc? I think, and there are 2 steps. Yeah, zinc, sorry. Yeah, zinc. And there are 2 steps. It's this one over here that's inhibited, and I think, which other one? Is it this one, dehydratase? Yeah, okay. Okay, good. So those 2 are, if you have lead poisoning, it inhibits zinc, you need zinc for these 2 steps. So know that. And then I just kind of went through each, oh, there it is, Jesus, okay. Yeah, zinc, inhibited by lead, and then over here, same thing. Okay, so then this next step down here, if you have this HMB synthase deficiency, that's probably what they'll call it on the test, but the notes had a different name for it as well. You want to know what's elevated. The substrate prior to the enzyme deficiency will be elevated, and you want to know the clinical symptoms. So this one is very characteristic because you'll get this the change in the urine, right? So it gets oxidized again. It has like this this dark appearance to it. What's important here is these patients are not photosensitive, right? So that happens in the next 2 diseases we're going to talk about. Okay, yeah. Why is that? Oh yeah, so ALA has a structure similar to GABA. So barbiturates, they will upregulate these GABA receptors. So if you have, since ALA is very similar in structure, it will compete for it, and that could be very problematic for people with this AIP. Okay, so just keep in mind, I don't know that they'll ask you, but if they do, barbiturates are just, they upregulate GABA, so they're used for like anxiety and stuff like that. All right, yeah, and this is the next step. So now we're getting into the ones that end up being photosensitive, CEP. So you're going to get the increase of these substrates, and then now you're going to get this red urine, not darkened urine like the first one. This is actually red urine, and they are photosensitive. It's because the structures, I think they have a ring, the porphyrin ring. Yeah. Yeah, so that's what actually causes it. So CEP and the next one we're going to look at both have these abnormal porphyrins, and that leads to the development of this skin sensitivity to light. Okay, so the last one is PCT. Ooh, and it's very similar. The urine will turn red as well. You also get it. The one thing I remember, okay, so CEP will be in kids. So take note of that, hypertrichosis. So they grow a lot of hair. CEP will be in kids. PCT is acquired, I believe, yes. So their urine will turn red. But if you're looking at it and you're trying to differentiate, the age will be a good way to do that. Onset, yeah, so acquired chronic disease of the liver could lead on to that. So that's a good way to do it, right? And you can see these lesions are very sensitive to the sun. All right, so I put a star on this slide. Make sure you can understand these. So yeah, so B6, so we're going through the process, through the pathway, right? So B6 is that first step. Lead poisoning inhibits zinc, and then we go through the 3. This first one, AIP, just their urine will oxidize and turn dark, like black. And then CEP, photosensitive, red urine, kids. PCT, photosensitive, red urine, adults. Acquired liver failure type stuff. All right, bilirubin. So what happens, so bilirubin is a product in red blood cells. So if you have hemolysis, a lot of breakdown of red blood cells, you get buildup of bilirubin. That's why people that have such like G6PD deficiency, stuff that'll cause a lot of hemolysis, they'll get elevated bilirubin so that they can look jaundiced, or they get scleral icterus, right? The yellowing of the eyes. Now, what ends up happening is you get this bilirubin in the blood, right? And so it's unconjugated at this point. Once it gets to the liver, it gets conjugated, and then it gets stored in the gallbladder, okay? Then from that point, it can be excreted as bile and whatnot. And then most of it is reabsorbed, but it can also, it makes the color of feces and urine. So urobilinogen or stercobilin, urobilin or stercobilin. Okay, so we'll go through the different types of hyperbilirubinemia. When you think prehepatic, before the liver, you want to think about hemolysis, red blood cells lysing, okay? That's where all the bilirubin is going to spill into the blood because of all the cells that are lysing. Now, if it's prehepatic, that means it's not conjugated. It doesn't conjugate until it gets to the liver. So prehepatic is going to be unconjugated. So when you look at the labs, I'm pretty sure we have slides on it. When you look at the labs, you would expect an elevated unconjugated bilirubin. Once you get to the liver, it gets conjugated, so you would expect to see increased conjugated. Posthepatic is usually we're talking about some sort of stone in the bile duct, and that is going to be conjugated as well, right? Because it already went to the liver. Okay, so this kind of goes through the breakdown. Right. So look, this is important. You need to be able to identify this. So what would you expect in these patients like G6PD, the thalassemias, anything that's going to cause hemolysis? You're going to increase total bilirubin, of course, but which of these specifically? Serum unconjugated bilirubin. That's the main thing you're going to worry about. Why is it not in the urine? Well, because it has to be conjugated before you can get it to the urine. So if it's not getting to the liver, then it's not going to be conjugated, so you can't process it through the renal system. All right, so hepatocellular. Now we're talking about intrahepatic, so liver problems. So you can see this bilirubin, look at this guy's eyes, right? So now you're going to get both unconjugated and conjugated. Now the problem is that with hepatocellular problems, here we go. Yeah, so it's unconjugated, but once it gets to the liver and it's conjugated, it's going to be elevated, but you also have trouble getting it into the liver. So it's a liver problem. You have a problem getting the bilirubin into the liver, but what is actually in the liver gets conjugated, but it's not processed correctly. Okay, so the correct process would be to excrete it through the intestine, through the bile, so that it could either be reabsorbed properly or go through the urine or the feces, but it's not. It's like regurgitated back through the diseased liver. Okay, so you're going to get both the increased unconjugated and an increased conjugated. Okay, so again, you're going to get it to the liver, but it's not going to be able to process it properly. So it'll be unconjugated, but some of it will be able to be conjugated, but it's not going to be regurgitated back into the bloodstream. Okay, so the point being is that if you had intrahepatic increased bilirubin or jaundice, you're going to get an increase in both. So I'll try to say it one more time, make sure we get it straight. So you are going to be able to get it to the liver. Okay, not all of it's going to be able to be conjugated because it is a diseased liver. Some of it will be able to get conjugated, but instead of being processed through the bile like it should be, it's going to be regurgitated back into the blood. Okay, so you're going to expect to see both. All right, so this is going to be super important. And again, you can see some is in the urine because some of it was conjugated, just not the vast majority of it's going to be regurgitated back into the blood, which is why you get this increased conjugated. Now, of course, if you saw this, you would expect to see signs of liver failure. Okay, so ALT and AST because it's a liver problem. Okay, now posthepatic means you have some sort of blockage. So you're not going to get any stercobilin, so they'll have pale feces. Now this should make sense because if you have a blockage there, then it can't be processed. Then the bile can't get out, and if the bile can't get out into the GI tract, then it can't be processed correctly into the feces, right? So it's going to be stuck in the bile duct. So they'll say they have pale colored stool. That's classic for some sort of posthepatic or obstructive jaundice. The bile's the bile with the bilirubin in it is blocked in the gallbladder. So can't absorb fats or soluble vitamins, again, because that bile that's blocked plays a major role in being able to do that. Okay, so again, the process, the liver's fine. So you take this unconjugated bilirubin from the blood, you put it in the liver, you conjugate it. Now you're going to get it into the bile duct, but since there's a blockage, the only place it could go is to be regurgitated back into the blood. Okay, so it's not, you can see this line here. This is the normal process it's supposed to go through, but it can't because it's blocked. So the only way is pathologically, it's going to get regurgitated into the blood. And so you're going to get conjugated. So now, if you look at this, you could see what would we expect? Well, the liver's fine. So we're able to take the unconjugated and conjugate it. But now, since we have a blockage, everything's being regurgitated back into the blood, and you get an increased conjugated bilirubin. Now, what would you expect on the lab values? Well, since it's the blockage of the gallbladder, you would have an increased ALP and GGT. All right, now this is normal physiological jaundice of the newborn. You can put them under fluorescent light if need be, but it's just a normal process. When you're born, you have this low UDP glucuronidyltransferase, okay? So the unconjugated bilirubin is increased because this is the enzyme that'll help to that conjugates it. If this is not, if it's bad enough, like you don't actually end up making this enzyme, you can end up with kernicterus, which is like you can see here in the basal ganglia. And it leads to some issues, some intellectual problems, and some motor issues called kernicterus. All right, so Crigler-Najjar syndrome 1 is the bad one, yes. So you don't have this UDP glucuronidyltransferase. So you get unconjugated hyperbilirubinemia. Why is it unconjugated? Because this is the enzyme that conjugates it. So if you don't have that, you're going to get increased unconjugated. Type 2, you have a little bit of glucuronidyltransferase. So it's not as bad, but you're still going to get elevated unconjugated bilirubin. This is actually responsive to phenobarbital. This is used for seizures. No idea why it works. That induces the enzyme somehow, how it works, but it'd probably be good to know that. But obviously you can't use this in type 1, right? Because there's no enzyme there. It works on this because there is a little bit of enzyme, 10 to 20%. So this is upregulates it. Gilbert syndrome, or Gilbert, I don't know. So this is about 50%. So this is the mildest form of it. Again, you're still going to get unconjugated bilirubin elevated. All right, so I just put this in here. This is severity of it. So age is a good factor. So type 1 is very young. Type 2 is a little bit milder. So you're a little bit older, and Gilbert, maybe you'd see older if you see it at all. And very importantly, liver enzymes are normal. So you get a normal AST and normal ALP. I'm sorry, normal AST and normal ALT. AST and ALT, yeah. Right, but and the whole point is that we are lacking this UDP glucuronidyltransferase, okay? But it's not, the liver's okay, you're just missing this enzyme. All right, Dubin-Johnson syndrome. This actually calls what this is black liver. It's a problem with the ABC transporter. Now, this is the one that's different. You get elevated conjugated or direct bilirubin. Direct is the same thing as saying conjugated. So this is the one that deals with conjugated, okay? And again, liver enzymes would be normal. All right, we kind of know these, right? We know our liver enzymes. Remember, AST to ALT 2-to-1 ratio would indicate some sort of alcoholic disease, a shot of tequila, right? But typically hepatitis, other problems, you get ALT greater than AST. We know this, if you have an elevated alk phos and a GGT, that would be some sort of gallbladder problem. And then, yeah. All right, good. Okay. Just problems that can end up causing increased bilirubin, but we kind of went through those, the ones you need to know. Right, we talked about that, Right, what is ascites, right? Remember, the liver has to make albumin or proteins, right? And the albumin stays in your circulation. So if y'all remember from renal, You have hydrostatic pressure that pushes out, and you have that oncotic pressure that holds stuff in the vessels. Albumin is the major player for that oncotic pressure. So if you have liver failure, you don't make albumin, so there's no oncotic pressure there. So all of the fluid goes out of the vessels, and when it goes out of the vessels, it gets here. So whenever you see ascites, same thing with those kids that have Kwashiorkor's disease. That's a protein malnutrition. They don't have albumin, therefore they don't have oncotic pressure. All the fluid goes out of the vessels into the interstitium, and they get ascites. Ooh, are we going through all of these? Oh, okay. Yeah, so you would expect, right? So this would be this is your albumin peak. So if it's decreased, like you would expect to see ascites in this person, just because of it. So this could be a sign of liver failure or some sort of protein malnutrition. And you get this this bridge here between these two. This is the gamma peak from your immunoglobulins and this beta. So there's some sort of bridge. I'm not exactly sure why that happens, but it just is a sign that that there's cirrhosis involved. All right. Yeah, so I don't know that this is really going to come into play, but of course the liver makes clotting factors. So if you have liver failure, you have problems clotting. So you're going to get bleeding issues. So we know factor 7, the extrinsic pathway, and then all the other ones go pretty much through the intrinsic pathway. So we're not going to go through all that. Ammonia, if you can't process it properly, like to make urea in the liver, all that nitrogen builds up and you can get problems. So either a liver problem or a kidney problem, you can't get rid of the urea. Well, if it's a liver problem, you can't make urea, so you get elevated nitrogen. If it's a kidney problem, you get increased urea because you can't get rid of it. Either way, it's not a good thing. All right, we talked about this. Yeah, so right, so anything that's foreign to the body is tough, like a xenobiotic. So you know, pharmaceuticals are xenobiotics for the most part. Yeah, and then we go through the phases, but we've talked about these, all the P450 enzymes. Let's see. Yeah, I mean, y'all talked about this in FTM as well, in inducers versus inhibitors. So it has a lot to play. Certain drugs like warfarin are very much affected by these. But ethanol uses the CYP2E1. So it can actually interfere with metabolism. Stuff like acetaminophen. Right, so typically acetaminophen isn't toxic, but this NAPQI can be toxic. If you take it in normal doses, this is negligible, but if you overdose on it, this becomes a problem. Excuse me. So if that's the case, that's when acetaminophen becomes problematic, and you use N-acetylcysteine as the antidote for that. Yeah. All right, and then alcohol. See, I think, is it this one's okay, right? This is the one that becomes, yeah, high doses. Okay. Mitochondrial acetyl, yeah, this is fine, but if you drink a lot of alcohol, then you get this cytosolic acetaldehyde, and that's problematic because the acetate's released in the blood. I can't really remember too many questions that came from this, so. Right, but this MEOS, this microsomal ethanol oxidizing system, that's what they talk about here. So this is problematic if you drink too much alcohol, right? This acetaldehyde is toxic. Okay, here again, this is the one that's problematic, cytosolic aldehyde, right? So this acetaldehyde will accumulate. Obviously, that's toxic to the body. Y'all may have heard this, this Asian flush syndrome. These people have this variant, so they're very sensitive to acetaldehyde. They'll get flushing and elevated heart rate and stuff like that. Right, and then remember, alcohol will block gluconeogenesis. So people will drink on an empty stomach because it's easier to get drunk. The problem with that is that once you're hypoglycemic, the alcohol will actually block the process of making new glucose anyway, so it makes it worse. In this process, what alcohol actually does is it increases that NADH to NAD ratio. So remember, when we talked about it, we said when we have elevated levels of NADH, we can't go through that Krebs cycle. that NADH is going to push you towards ketosis. So this is another example. Elevated NADH will stop the process of the Krebs cycle. But here also, it will also stop the process of gluconeogenesis as well. Okay, so you're not mobilizing, you're not using energy there. Right. Y'all probably heard of disulfiram. It is used for people to stop drinking. It blocks acetaldehyde dehydrogenase. And what that does is that makes you feel like you're hungover immediately. You can't break down alcohol properly. All right, so when people used to overdose on stuff like methanol or ethylene glycol, they used to give them ethanol to actually block it because ethanol is a competitive inhibitor, but now they actually can use fomepizole. So you need to know this slide, you need to know this process, you need to know that you can use fomepizole to prevent the formation of these bad products over here, okay? And yeah, so, you know, they used to make moonshine or still do down south. Yeah, it can cause blindness. So be aware of that. Ethylene glycol is antifreeze. So it's problematic because it tastes sweet. So sometimes kids or pets will get into it. And yeah, so again, if we go back a few slides, this is the treatment for it. Give fomepizole because they're competitive inhibitors. You're trying to override the process of these being broken down. If you can give something else to use the enzyme, then you're not going to break these down as fast. All right. And then you get a high anion gap metabolic acidosis. So be aware of that. These vitamins, we'll go through these quickly. Okay. A, D, E, and K, y'all know, are fat soluble. This is just basic memorization. So like what does what? Vitamin A, of course, vision, you get night blindness. If you're deficiency, that's probably what you want to be aware of. These pitted spots are in the eyes. Also, vitamin A is a antioxidant, so the immune system can be affected. A, C, and E. So ACE, they're all used as antioxidants. Hypervitaminosis doesn't happen very often, but you can get increased intracranial pressure. Y'all know about vitamin D by now. Yeah. So of course, hypervitaminosis will do the opposite. You get a whole lot of calcium and phosphate. This could lead to calcium stones, kidney stones. Right, vitamin K, of course, is used in this process of clotting. This gamma glutamyl carboxylase, or gamma carboxylation process for vitamin K is used for the clotting factors 2, 7, 9, and 10, I believe. And warfarin obviously blocks vitamin K epoxide reductase. When it does that, you don't have vitamin K, so you can't go through gamma carboxylation. All right, neonates, when they're born, they are given an IM injection of vitamin K because what actually happens, the GI tract, the bacteria in the GI tract help to make this vitamin K. So it takes about a day or so for your gut flora to be developed, or a couple of days, I'm not exactly sure. But they give them an injection right when they're born so that they don't have problems bleeding. All right, so of course, any deficiency, you can have problems clotting. Vitamin E is also, like we said, A, C, and E are all antioxidants. And then the water-soluble ones, you know, vitamin C, scurvy. B1's important when we talk about Wernicke-Korsakoff syndrome. That's the primary one that we talk about. Remember, it is the T and TLC for Nancy. So any of these enzymes, the 3 that we talked about, will all be affected. And remember, maple syrup urine disease uses that branch chain keto acid dehydrogenase, and that uses thiamine or B1 as well. All right. Beriberi, wet heart. So the heart is wet beriberi, the brain is dry beriberi. Okay, so those are problems. Look at the pitting edema here. And here's the Wernicke-Korsakoff. So the Wernicke component happens first. That's where you get blurry vision or ophthalmoplegia and ataxia and confusion, right? That triad. Then later on, you end up developing this Korsakoff, and that's when they start with the confabulations, right? It like blocks your anterior grade memory, so you have no connections there. So what this means is they tell stories genuinely thinking they're true, and they're just completely made up. But that's pretty far advanced. One of the questions, interesting questions that I've seen about this is, so an alcoholic goes and he has a thiamine deficiency, right? He has like Wernicke-Korsakoff, and he went to a party or something and like ate a piece of cake, and then like he went into like he ended up getting this really bad like these neurologic complications. Maybe he went into a coma or something like that right after. And it's like, well, what happened? Well, the problem is when he ate the cake, it was full of sugar, so he had to break down all of the glucose, and you need thiamine as a cofactor. So the little bit of thiamine he had in his body, he just used up eating the cake, and ended up, yeah. So if you've seen that question, or maybe it'll come up, but it was confusing because you're like, well, he doesn't have it, but they have low levels of thiamine. He ate glucose, he used the rest of it up, so this got really bad. So just be aware of that. Riboflavin, I think we said earlier, that would be your glossitis. There it is, and chelosis, that's in the sides right here. Niacin, we said the three, yeah, dermatitis, diarrhea, dementia. Heart and lung disease, we talked about that in relation to niacin. Carcinoid syndrome is the development of serotonin, too much serotonin. So that conversion there. B6, I don't know if they ask anything about this. Just be aware that things like isoniazid will block or will use up all the B6. So like when we talked about that ALA synthase and the heme synthesis pathway, that would be the thing that they ask about B6. I don't think they'd get into any of this. Okay, this either. Oh, there's your isoniazid. Yeah, so be aware, but that's a big problem. They have to be on, anytime you give isoniazid, you always give them B6 supplements. Biotin carboxylation reactions. That may be something that they talked about. This is in Avidin is in these raw egg whites. It inhibits the absorption, so that may come up in regards to biotin. Folic acid, B9. We know megaloblastic anemia. That's what will happen, and we know that neural tube defects, yes, in kids, right? So moms have to be on folic acid supplements. B12, we talked about this already. Megaloblastic anemia, you also get neurologic deficits, and methylmalonate or methylmalonyl CoA right here. That will show up in the urine, immediately diagnostic for B12 deficiency. We talked about pernicious anemia, right? Blocking these parietal cells, you can't absorb B12. All right, again, we talked about this. Neuropsychiatric symptoms, that's B12. There's your macrocytic, big red blood cells. And also, sorry, this is the one they usually talk about, this macrocytic anemia, but you also get these hypersegmented neutrophils. Okay, so I doubt they give you a question about it and not mention this, but don't let it confuse you, because this also happens, this hypersegmentation. Remember, you're supposed to have a maximum of 5 lobes, right? But this is more, so that just happens as well. All right, what is this? What is this? I don't know. I guess, well, if you have questions about anything, we just answer questions later. All right, lysosomal storage diseases. Fortunately, these enzymes are hard to remember, but you kind of need to know these as long as you kind of know the symptoms that are involved. It's one of those things you just have to sit down with and understand and just kind of work through. But there are certain things that are very characteristic. I'll try to, let's see. Well, let me just go real quick to the end. I'm gonna show you something. This. I'm pretty sure that y'all had, does this look familiar? Did y'all have this in your notes? Dr. May?
Yes.
Okay, this is really good. Okay, so if you know this, you could probably answer all the questions about this. So this is a good chart to look at when we do that. Okay, so anyway, so hurlers and hunters, they're very similar in presentation. They just have small differences in, remember, hunters can hunt because they can see, right? No corneal clouding, whereas hurlers, they cannot. They have the corneal clouding, right? So they can hunt. So to hunt, you can't have corneal clouding. I guess that's how you remember it. Okay, and it's X-linked, X to hunt, to target, right? Tay-Sachs, fairly common, not fairly common, but it's compared to the rest of these, it's pretty common. So yeah, just be aware of this. You get those cherry red macula that you could see here. That's characteristic for it. Also this, I highlighted this as well. They could put this in words or show you a picture of it. That's fair game, this onion shell inclusion, okay? No hepatosplenomegaly. That's different from some of the other ones. Gaucher's disease, I remember them giving us a question about this Erlenmeyer flask formation, and they were talking about Gauchers or whatever. They were talking about this. We had this. They asked somebody had bone problems. So this Erlenmeyer flask formation, that's just this shape right here. So the bone is kind of tapered off right there. You can also get this crumpled tissue paper. That's fair game as well. They could ask you that. Fabry's disease, no hepatosplenomegaly as well. I don't know what they would ask about this one though. Niemann-Pick, same thing. Also get cherry red. I think the trick is if it has a hyphen in it, you get cherry red spots. So Tay-Sachs and Niemann-Picks. Right, again, look how big. The spleen, the liver, hepatosplenomegaly here. So different than Tay-Sachs. Remember, Tay-Sachs, no hepatosplenomegaly. Niemann-Picks, you do get hepatosplenomegaly and foamy cells. So that's a good buzzword for that as well. I don't know that they'll ask about this one. Pompe disease, remember we talked about the heart problems, cardiomegaly. ISL disease, inclusion cells. So remember they can't mark the cells for, or the lysosomal enzymes yeah, they don't have this mannose-6-phosphate marker. Okay, so these enzymes can't be tagged to go to the lysosome, so they end up in the bloodstream. So this is a good buzzword, this mannose-6-phosphate. I know y'all talked about this in FTM if y'all remember. So if you don't have that, all of these lysosomal enzymes that are very, very active, very strong, you know, because they're supposed to be in the lysosome breaking down stuff, they end up in your bloodstream and it causes a ton of problems. All right, so see that here. I put it, I don't know why. Maybe they put this on our oh, you can see the inclusion cells. That's what those are. Okay, yeah. So interesting. Yeah. So why are all these inclusions in here? That's because all of the lysosomal enzymes that were supposed to be tagged to go in the lysosome to break all this stuff down, they're in the bloodstream, right? Because you didn't tag them with the mannose-6-phosphate, so you can't break down any of this stuff. That's why there's these inclusions. So yeah, so that's fair game as well. All right, and like I said, this is a great little chart to learn everything. And that being said, we're done. Do you guys have any questions? All right, if not, good luck on your exam. I'm sure y'all will do great. Trust yourselves. You know more than you realize. Yeah. All right, I'll stop the recording. I'll try to have this posted. It'll take a while for the process, but yeah, good luck, guys.